Muy buenas tardes a todos. Buon pomeriggio a tutti. Welcome. Mi nombre es Elena García Guerra y como vicedirectora de la Escuela Española de Historia y Arqueología en Roma, centro perteneciente al Consejo Superior de Investigaciones Científicas de España, os doy mi más cordial bienvenida. Benvenuti. Y con este breve saludo quiero en primer lugar agradecer públicamente a los coordinadores el trabajo y el tiempo dedicados para celebrar aquí este importante seminario y en segundo lugar agradecer también a todos los ponentes el esfuerzo por su participación, ya sea aquí en la sala, ya sea en presencia, y también un saludo para aquellas personas que nos sigan en directo por el canal de YouTube. Por otro lado, desde el punto de vista institucional, estamos muy contentos de la colaboración con la institución Milà i Fontanals, a la que pertenecen Marta, Portillo y Aroa García-Suárez, y de que la actividad se inserte y haya obtenido financiación de la red Conexión Arqueología-CSIC, que tiene entre uno de sus objetivos fundamentales fortalecer la colaboración entre los diferentes centros y el intercambio de conocimientos entre los diferentes grupos que a ella pertenecen. He comprobado que en estos 2 días vais a abordar la aplicación de las técnicas microarqueológicas a muy diferentes entornos, ya sea desde el punto de vista geográfico, ya sea desde el punto de vista cronológico, lo cual da muestra de la seriedad del planteamiento científico y de la importancia de los objetivos que subyacen en este seminario. Por tanto, y dado que tenéis por delante un programa muy intenso y muy concentrado, no me queda más que desearos buen trabajo, buon lavoro y una buena estancia en Roma. Muchas gracias a todos.
Thanks so much, Elena, for this wonderful welcome, institutional welcome. As I said, well, I really speaking on behalf of my colleagues, the coordinators of this conference, colleague Aroa García-Suárez at the Spanish National Research Council in Barcelona, and Juan Francisco Gibaja, and Antonio Pizzo from the Escuela Española de Historia y Arqueología en Roma. As said, this conference aims to review the current methodologies, trends and applications, and advances in the study of the microarchaeological record to address the challenges of integrating high-resolution approaches, and to evaluate the contribution of experimental and ethnoarchaeological comparative records, to discuss the role of microarchaeological records in exploring major research questions in archaeology, and to suggest possibly future venues and directions. So we are very grateful first to the Spanish National Research Council here in Rome for hosting this international conference, for all the participants for sharing with us research, and for all attendants here in person and online through the YouTube channel. We are also very grateful to the Archaeology Hub for the finding, and also this conference organizing the framework of ongoing Marie Curie project. So just very quickly, this is a hybrid event. So this conference will be fully recorded and will be able to watch with the YouTube AR channel. We have just last minute change in the program. Our first speaker will be Cristiano Nicosier from Università di Padova. So, Cristiano, are you ready to go? Thanks so much for making all the efforts to be here today with us. And this session will be shared by my colleague Aroa García-Suárez. So, thank you very much. enjoy the conference.
Okay, thanks for inviting me, and thanks for being so patient to move my talk at this unusual time. So I hope it will be a good talk. And I will speak about geoarchaeology of wetland contexts, which is mostly focused on Bronze Age wetland contexts. And basically, I saw later the type of conference and the topic of the conference, and so I will really try to focus mostly on microarchaeology or micromorphology as we know it. And so basically, I will start by saying maybe not everybody knows what soil micromorphology is, although many very good colleagues are here in the program. But basically, micromorphology means to study undisturbed blocks of sediments that or soil that we collect somehow from exposed stratigraphic profiles, mounted on glass slides that we know we call thin sections. So it's a very thin slice of this soil, and then we study this thin section in transmitted light at high magnification in under the petrographic or polarizing microscope. And so basically, I will try to focus on the micromorphology in geoarchaeology in wetland contexts. And I think it's important to remember that micromorphology is one of the scales of observation in geoarchaeology, and I consider it a part really of archaeological excavation in a way. When there is a good connection and flow of information between excavation and the microscope, then micromorphology is very useful. If there is no flow of information between the microscope and the excavation, then micromorphology can be really self-resilient and creating more questions than answers. So basically, I start with three aspects of wetlands that are relevant for micromorphology in wetlands. First of all, the first aspect is not peculiar to micromorphology, but is the fact that in wetland environment, we find wetland sediments, which is a made up label to a variety of environments that go from coastal wetlands to intermorenic lakes, to large lakes, to smaller mires and pit bogs and fens, etc., etc., etc. So there's a variety of environments, but the point in these environments is that we find all the type of sediments that are known to occur on the surface of the planet, like clastic, organic, chemical, and anthropic. And we are very interested in the anthropic sediments, of course. And in the point, what is interesting in this type of in micromorphology of this type of environment is that there is, in geoarchaeology more generally, is that there is a very straight link between the type of sediments we find in wetland sequences and the environment where they form. So we have a very high potential of basically informing us on past sedimentary environments and sedimentary evolution. But that's not the main point. The second point is that we basically have, as everybody knows, in wetlands, we have the preservation of organics because decomposition is slowed down or hampered, and so at various degrees. And so we have, of course, from pollen to macro remains to whole bodies or wooden objects, they are preserved in wetland environments. And what does this mean for micromorphology and geoarchaeology? Well, it means that we have a lot of information out there. But I think in a way for micromorphology, for example, the point is not necessarily to be able to identify each component to the botanical level, because anyway, oftentimes we lack the statistical relevance of the identification. We identify 1, 2, 3 bark fragments and 2 roots fragments in a thin section. So that's not really the point of micromorphology. my opinion. And so there is another characteristic that is even more interesting for micromorphology and for geoarchaeology in wetland environments, and it's the fact that in wetland environments, bioturbation is very, very, very low or zero or close to zero. And this, in my opinion, is the key characteristic that makes wetland environments very interesting to be studied with micromorphology. I chose some images here to show contexts that are absolutely not wetland contexts, where we see that bioturbation by earthworms digging pedotubes, bioturbation by dung beetles, or total homogenization like what we find in urban deposits, or so-called dark earths. Okay, they completely re-homogenize the soil and re-homogenize the deposit, and this really makes a difference from seeing an archaeological thin section from a wetland site, a waterlogged site, or a predominantly wet site, and seeing a thin section from a non-wetland, non-waterlogged, like, let's say, oxidized site. This is the main difference in my experience. And I chose this image down here to say, yeah, bioturbation is one of the soil forming processes, but also other soil forming processes will impact on the deposits in non-wetland environments. And the main difference is that bioturbation acts very fast. Some pile dwellings being excavated the moment you lower the water table, in that moment bioturbation will start, and degradation of wood and a lot of other processes. Soil processes act more like on, let's say, longer time scale, geological time scale, and namely the main process is leaching of carbonates and decalcification of the profile, which brings about disappearance of ash, for example, and other processes. But it's actually much slower than the impact of bioturbation, which starts immediately as soon as the sediments are made available. So with these three aspects in mind, I tried to show you two case studies of wetland sites, of Bronze Age wetland sites that were particularly interesting. And the first one is the site called Oppeano via Isolo. Why is it a wetland? Because it sits in a lower reach of the Adige floodplain south of the city of Verona. Okay, these big orange surfaces are the last glacial maximum alluvial fan, which have a rubified alphisol, and actually this picture comes from that surface. That's why I colored it in orange. Okay, and then you have these incised valleys, incised by rivers, like in the early Holocene, and downcut their course and built extensive lowlands where sedimentation is fluvio-palustrine and the water table is very close to the surface. And this site was excavated as part of a rescue archaeological project, and you see for the construction of a pipeline in 2015, you see this is the site with well points working, and this is the site with well points not working. So in one night, the site basically is completely filled with water. Okay, the water level is very close to the surface here. And so this is when they stole the gasoline for the pumps during the nighttime. The pumps didn't work anymore, and in one night, the water went to basically close to the surface. This site is an early Bronze Age. One site has been excavated in a long and narrow trench following the pipeline. The trench is about 80 meters long and 6.5 meters wide. And what is interesting is that it revealed the remains of the base, let's say, the base level of 8 huts, wooden huts of the Bronze Age, and also of other structures like palisades and other liminal structures. So this image you see is a summary of all the phases. So it's not just one phase, it's a summary of all the structures that are not all contemporaneous, but they are roughly contemporaneous, okay? And the degree of preservation is the one that you see in these images. These are Middle Bronze Age 1 walls, let's say, with wattle and post technique. that describe the perimeter of these structures, of these houses, and in some of these structures, we even had elements of the, we would say in Italian, the alzato, so the wall itself really clearly visible. You see what is the size of the wooden elements that were recovered. More than 1,000 wooden elements were recovered, and so floors, okay, floors made with pedorelicts of the rubified alphisol, sometimes with local calcareous silts, and fireplaces very well preserved, stacked upon each other inside these structures. But what is mostly interesting as for a micromorphologist is the preservation of the internal stratification of these huts, and the stratification is not bioturbated, so you can really follow from one side of the house, or let's call it house, from one side of a structure to the next, each individual lamina or very fine stratum, and all the organics in this stratum is preserved. So not only the organic is preserved, but there's no bioturbation, which would have totally re-homogenized this material. And this is, we are very familiar with several Bronze Age sites. In dry contexts, you do not find finely laminated sediments because they have been thoroughly reworked by bioturbation. So this is part of the thesis of a student of mine, Federico Polisca, PhD thesis, and he is working on deciphering. We are working together, deciphering each individual lamina, let's say. What does it tell us? Okay, so of course we did thin section. Thin section contained each thin section a number of subunits. Each subunit was assigned what we call a soil microfabric type, because several times you get the same material over and over again, and so you need to sort of make groups. And the main point is that these are basically Bayer houses. Bayer houses because what we see under the microscope is levels of ash from the fireplaces, wood ash, never a single burnt spherulite, so it's not burnt dung, is wood ash spread on the living floors, let's say, intercalated with, this is a pseudomorph of oxalates testifying wood ash, intercalated to herbivore dung, very rich in phytoliths, very rich in spherulites. And so this is an alternating sequence that we don't see only in one thin section, only in one trait, only in one point. This is the style of the sedimentation, we would say, in two of these houses, the two that we studied this far, repeated over and over and over: wood ash, dung, wood ash, dung, wood ash, dung. So these are solid evidence for Bayer houses, because in these houses, how do we know they're not just Bayers? Because there are fireplaces, there's pottery, burnt bones, loom weights, material related to weaving. So the typical inclusions that we associate with domestic structure, but strictly intercalated to repeated accumulation of dung. And so basically we have here two Middle Bronze Age one Bayer houses, and now we are trying to understand what type of herbivores and where the herbivores spend there all year long or not. So it's work in progress, but I think it was an interesting example to show you. Second example, we go back in time. The Lucone pile dwelling is early Bronze Age pile dwelling, located again in a wetland that form into the, let's say, the wrinkles of the moraines that border the southern limit of the Garda Lake. So these are LGM, last glacial maximum moraines, and they are wrinkled. And so in these up and downs, there are topogenic basic mires, small pit bogs, where water drains through the glacial till until it finds impermeable layer, over consolidated till, or more clay rich layers, and drains laterally. and basically creates a small mire. And these were sites that were inhabited in the early Bronze Age in Italy and were very famous pile dwellings like Lavagnone and Lucone were located. And these are excavations led by Claudia Mangani and Marco Baioni. Wood is preserved, okay, very important, very nice. But for example, you see the midden hips, okay, are finely laminated, and they are finely laminated because there is no bioturbation that intervened into homogenizing them. And we notice that there is colonization by earthworms and ants during the excavation almost. So at the moment you lower the water artificially, bioturbation will start. It's like a time bomb, okay? These midden hips, we know now that they are midden hips, but the first questions were like, are they structures? Are they fireplaces? Are they structures that were carried out, activities that were carried out in situ, or are they an echo of something going on somewhere else? And you see the laminated light colored sediments, we see it very well here, and darker colored sediments here. And I start from the end in a way that what they are really basically discard of hard maintenance material outside the platforms. And this is a very nice image from Petrecan work, from ethnoarchaeological work. So they're just midden heaps, waste heaps, but they contain a lot of information for us, because as archaeologists we like waste, we like garbage. And you see in thin section, these heaps are perfectly laminated. There is not one single channel, not one single pedal tubule, ruining our nice lamination of the layers. And remember, the idea was that we had the light color and dark colored layers. The light colored ones are basically mud plaster that were used to plaster the surface of the fireplaces. And if we look at those materials under the microscope, we see that they contain pedorelicts, so broken soil fragments, and these broken soil fragments contain clay coatings, and clay coatings, they tell us these are soil that have a BT horizon, so an argic or argillic horizon, and there are not many soils that have this type of horizon around this area. Specific types of soil that form on the carbonated till, so that had a long time to develop since the beginning of the Holocene, are these red soil that are known in the area as ferretto, which means ironish soil because of the color. So they were quarrying this specific material mixed with more calcareous material underneath to create their plastering of the floors, and periodically dismantling them and dumping them. The dark colored layers are instead ashes mixed with a lot of organic material, plant remains mostly, and we will see a little in a second what they are. And what is interesting is that in this site we observe that everything was burnt as a combustible. Chaff was burnt, wood was burnt in large quantities, grasses were burnt, because we have vitrified phytoliths and blackened phytoliths and phytoliths, but even dung was burnt in this site because we have blackened spherulites, and we know that blackening occurs when spherulites are burnt under certain conditions and in certain situations, basically. So really have a polycombustible economy in the early Bronze Age in this site, whereas for example in Opeano, the site we saw before, we haven't found a single indicator of burnt dung in a lot of thin section that we have been studying. The lack of bioturbation allows us to do a very precise lamina by lamina study of the activities that are at the base of the formation of these heaps. And if we think that not very often, actually almost never, the platforms above where people lived in pile dwellings are not preserved, these are the only echoes of what people did in their own houses, basically. And so this very precise lamina by lamina, layer by layer study is bringing together now micromorphologies, as we saw, but even phytolith analysis, for example. In this case, phytolith analysis is performed by Marta Dal Corso, who's now a postdoc here at the University of Padua, and this allows to very closely match the signal of what you see in a smear site or a sample for phytoliths, what you see in micromorphology, and then trying to link this to the bigger picture, to the field stratigraphy. And so I will just conclude my talk showing the fact that within the framework of this ERC project that just started, we are working on several Bronze Age sites. Of course, they are not all wetland sites. Actually, the minority of them is wetland sites. But comparing dry oxidized deposits with wet deposits, which are the best, let's say, degree of preservation, we hope to extract more information from Bronze Age stratigraphy, and specifically from Bronze Age domestic context, so from houses. And this will be done in these sites that participate to the project. And so I hope in the following years we will be able to provide some more interesting data on this type of Bronze Age domestic context. So thank you very much.
Thank you very much. Thank you very much, Cristiano, for this really, really interesting talk, and congratulations on your ERC project. I'm really looking forward to your results. I would like to remind you that we are broadcasting this through the school's YouTube channel. There's a few people connected, and I would like to encourage them, if they have any questions, to feel free to post them in the chat, because we should be able to receive them. There is a bit of discussion at the end of this session, but I think Cristiano has to leave soon. Please correct me if I'm wrong. So
yes, unfortunately, yes. Yeah.
I would encourage if you have any questions for him, you should formulate them now, both to the people here in the school and to those online. Are there any questions? Yes, Juan.
Despite Oppiano with all the uncarbonized wood, there are dendrochronologies involved.
Ashman.
Yeah, I don't hear the question.
Yes, no. Yeah, that's what I was going to do. Yes. So there's been a question about the site of Oppiano from Wellnoon regarding, because she's in there's a lot of wood, and she's asking whether there is any dendrochronological analysis going on or planned.
There has been a very small study done so far, because the woods are, I think, 1,500. Of course, they're not all datable, but some are quite big diameter woods, and we are looking for the money to making a more thorough study. But indeed, that will be a very key aspect of this site, because we need to know, are the huts really contemporaneous, or are there small changes that we didn't catch with the stratigraphy while digging? So there's a little bit of dendrochronology done, but we hope to do much more in this year, very soon actually. So if we get the money for it, basically.
She says thank you. Are there any more questions for Cristiano? ¿Tienes una pregunta?
Yes, my name is Marta again. Just a quick question. What do you mean by vitrified phytoliths? I saw that you one of your contexts, Lucarini site, and you mean high temperatures? Can you tell something else?
Can you hear her?
The question about vitrified phytoliths, I think
Yes.
what we mean. Vitrified phytoliths, we find them often in fireplaces, basically, because the vitrification is basically the melting of silica due to the presence of fluxing agents in ash, like potassium normally, and that lowers the temperature necessary to melt silica. And so you get these, they are called bubbly scoria, vitrified phytoliths, molten phytoliths, but they are basically very common in fireplaces, and they are basically favored by the lowering of the necessary temperature to melt the silica, which is brought about by the presence of large cations like potassium especially.
Thank you, Christian. Are there any more questions? I do have a question for you, actually, because as a fellow micromorphologist, sometimes I have incurred in difficulties sampling, and particularly sampling the type of context that I want to extract a block from. You know, regarding the particularly your first case study of Peano, I saw it, was it mainly horizontal excavation? I mean, how did you manage? Did you use strategic planes, or how?
It was a horizontal open area excavation, and it was also a rescue excavation. So the time was very fast. The timing was fast. So what we did was we left, I think in English they're called bulks. Like we left this testimone stratigraphico in Italian, we would say, like a small wall in the central part, sometimes two perpendicular to each other, and then sample those very carefully as the excavation progressed around us, and then they were dismantled at the end, basically, which produced very ugly plain photomosaics, but at least we were able to get you know, very precise analysis afterwards, after the excavation was done, of the sequence infilling such a structure, but huts. But yeah, we left basically a witness in the middle, or more than one witness, and then excavated around it. Yeah, the point is that once you saw the fireplaces, then you knew that you were more or less inside one structure, and so you knew how to go lower and lower, basically. But yeah, it was also dictated by the very fast pace of rescue archaeology. And so I don't know if this answers the question, but
Yeah. Thank you, Christian. Thank you.
Yeah,
so
no, my pleasure.
So no, I don't see any questions in the chat. So if no one else has a question for Cristiano, I think we can leave it here. Thank you very much again for joining.
Thank you very much for having me. Thank you. And sorry if I have to leave. Sorry, but thank you.
Our pleasure. Thank you so much, Cristiano. Thank you. Okay. I think he's still sharing his screen, and he's probably left. Okay, now here we are. Okay. So our next speaker is Niccolo Mazzucco, and he's going to be joining us virtually as well. Ciao, Niccolo.
Ciao, can you hear me?
Yes, I can hear you.
Okay.
Yes. So he's going to be talking to us about a multi-proxy experimental and archaeological case study from Neolithic harvesting tools from the lakeshore village of La Marmota, Italy. Thank you very much, Niccolo. The virtual floor is yours.
Thank you very much, and thank you, Aroa. Thank you, Marta, that invited me. And I'm sorry I'm not to be in Rome, but now I am excavating, so it's been quite a busy day. So sorry if I'm a little bit tired, but anyway, I will present this study, which is part of a larger project, which I working together with Juan Gibaja and Mario Mineo, so between the CSIC and the Museo delle Civiltà di Roma. restarting many different analyses on the record of a La Marmota site. So this site, probably most of you already know it, and has the site presented by the colleague Nicosia. This is a submerged site with a very good preservation of organic materials. It's located in the Lake of Bracciano near Rome, so I will not describe with too many emphasis the site, because I really think that this has been already presented several times, especially in the Scuola di Archeologia del CSIC at Rome. So as you can see, we have a lot of information of the site, mainly thanks to Mario Mineo, that together with the director of the museum, Fugazzola del Pino, has excavated the site for several years. So you can see we have some new datings. All this information is still unpublished, but is in course of being reviewed. So we hope that soon it will be everything available and published with all the details. So this is just a preview that we are going to give you. So you can see the site, the new datings on some caryopsids of domesticated cereal are between 5,600 and 5,200, which more or less confirm the datings already made on the wooden post. And so it is an early Neolithic village, not one of the earliest villages of Italy, but still an early village for the Mediterranean. And I'm sure it is, for what concerns the submerged site, is one of the most important Neolithic sites with such good preservation of archaeological artifacts. So one of the special finds of La Marmota is the sickles. These sickles were known since the first publication. Here I just show you a photo of one of the sickles that has been published in as part of the catalog of a very beautiful exposition, 7000 anni fa il primo pane, which is already, which has been done several years ago. And on this very beautiful object, these wooden sickles, there is not much published. And so one of the goal of the project was to restart all a series of studies on this object. Of course, there are many more. There is not just one complete sickles, but there is many complete sickles, and between complete and fragmented sickles, there is, I think, 52. And a good part of them still conserve the lithic insert inside the wooden with the glue. So this was a very interesting object, a quite complex object because it's composed of different components, different raw materials. And it's very interesting to have the possibility to study these tools, because they are part of a long tradition of tradition and innovation. And La Marmota wooden sickles, so you can see, are part of an evolution of harvesting tools that took place since the Natufian and then the Pipene in the Near East. And then it keep evolving during the Neolithic, the later phases of prehistory, and then, of course, Roman, medieval times, actual time, with a huge variability of harvesting tools, both from a chronological and geographical point of view. So in this presentation, we will only focus on 3 of the most well-preserved sickles. One of them, which in part it was already published, at least a drawing. It came from the layer, from the bottom layers of La Marmota, layer 2, and the other 2 comes from the upper layer of La Marmota, the middle, the one that correspond to the the most important occupation phase of the village. So you can see the position. The three of them are near or inside one of the possible house structure. And as you can see, we also choose these three because they are complete. We have all the three parts that compose the Sickle available, but as well there are three different typologies. You can see there is two main typologies, but here you can see we have this subcategory. So we have with this handle, open handle, then this very particular handle which has a T shape, and then this like a sword. So this you can see the details. So the measure of these Sickles, as you can see, they are quite small. They are between the cutting edge. It mainly is between 16 and 20 centimeters. So they are not huge object. In particular, these, even the handle is quite small. So I mean, if you have a very big hand, it's not very comfortable. So it's probably used by someone with relatively small or medium-sized hand. You can see this is the one with have the highest number of lithic insert conserved in it. And this, as you will see that more in detail, is very particular because there's a sort of decoration with some grooves all along the lateral ridge. And then the third one is this very enigmatic shape in the handle, in the base of the handle, that we have no idea actually of what exactly is the function of this T-shaped handle. So as you can see, they are very beautiful objects. The wood, we don't have all the details still about the wood technology because the study is in course by our colleague Laura Caruso, but we know that the wooden has been chosen, has been cut in some particular way to obtain this table-like blank, and then the object have been shaped, and you can see that they're well defined, they're well polished, so they are not just objects that were like quickly made or made in a simple way, but you can see there is a design in it. We also made, thanks to the aid of Miglia Monzota from the Institution Milai Fontanals of the CSIC, a series of scans as well for matter of conservation and preservation. So you can see here the detail of this, groove that you have on the lateral ridge. And what we applied is different techniques to try to approach the functionality and the technology of this tool from all the components. So studying the lithic insert with the technological and traceological analysis of the insert, the taxonomic and technological analysis of the wood handle, and then the analysis of the adhesive substance and of the residues that were possibly incorporated into the glue that has been used to fix the insert on the handle. So for what concerns the preliminary result from the work of Laura Caruso, you can see that two of them are made on Quercus, and one other is made on a Rosaceae malloideae species. These are species that were probably available in the surrounding of the site, and there are no then be exploited for doing craft work, even in other early Neolithic sites. For what concerns the technology and traceological analysis of the insert, so one of the questions was to analyze the micropolish variability of these tools, because as you can imagine, there can be a huge variability in the micro-wear polish associated with soil harvest. And so here you can see an example of a very smooth cereal plant harvesting polish, and another which is much more abraded and striated. So when you have two images, this is very easy to detect this difference, but when you have a lot of them, it's not easy to approach this variability. So that's why we approach these issues by using confocal microscopy. And so together with Juan Gibaja, with Fiona Pichon and Juan Ibanez, we started an experimental framework to do an analysis of the lithic insert and of the polish variability using a sensor-scan confocal microscopy that it's hosted at the CSIC at the Institution Mila Fontanals. The idea is to extract data, quantitative data from the polish, from the archaeological tools, and from some experimental tools to compare them and to see how statistically they can be classified, archaeological traces, by using the experimental one as reference. So we've been done, of course, using some polyvinyl siloxane impression, because it was not possible to put directly the wooden object inside the microscope or to move it from the museum. And so we worked on this impression, but this is something very common, especially for traceology. And this type of impression was recently demonstrated that were, in particular, the one we use, Provil Novo siloxane vinyl impression, were some of the most that are providing the better resolution, probably. So thanks to this impression, we have done a canonical visual analysis, usual study of the traces, so you can see each one of the insert with different magnification. As you can see, there is some insert that you can see that is impossible to study, because probably is the past insert that is being left from previous users. So one thing that we can already say is that not all the inserts show the same degree of development of the traces. So this is very clear in these sickles. You can see the first one has quite few traces, and the other are much more developed with a differential development. In some cases, this can be related to the position of the insert within the sickles, but such huge difference is probably related to the fact that probably the insert were replaced at different time. From the sickle were resharpened after use, but not all the teeth, not all the sickle, the lithic insert were replaced. Only a few of them that maybe were more abraded, or some of them were also as well possibly lost, not intentionally, which is very common when you are harvesting, that you lost some of your insert because of the heat, because of the stone in the soils. All this formation that we have on the use of the sickle is also possible thanks to an experimental program that we have, and now it's at least the third years that we will put this experimentation in the field. So harvesting different type of cereals in different locations. Here you can see me and Mario Mineo harvesting a Gentil Rosso, which is an old variety of Triticum aestivum that is cultivated in Toscana. As you can see, it's a very beautiful cereal, very high cereal. And so we harvested with a replica of La Marmota sickles, or one of the type. And we are doing this in different contexts in Spain. We are doing this in France. And so we are also comparing different factors that can influence the harvesting polishes, in particular climatic aspects, and as well the timing and the ripeness of the plant. So from here, you can see in this drawing that in the adhesive there is quite a lot of residues that are incorporated into the glue. So from the experimentation, we got the idea to analyze the adhesive to see what type of material was incorporated into the adhesive. And if we get a signal of the serial field directly from the serial field, so of the species that the tools have been harvesting. To compare the traces, we also integrated other experiment with sickles, always from La Marmota type, but as well from other sickle for cutting plants, for cutting other type of cereal, for cutting reeds. So to have a huge reference framework. And so then we started selecting different areas from the archaeological and experimental tools, following a quite standardized method for confocal microscopy. So choosing the most representative and the most clear spot to characterize each tool. And then we extract several texture parameters following the ISO parameters. And then from this, we obtain the values. And this is the result, a preliminary result, that you can see all the archaeological traces except for one teeth, but I will not enter in the detail of this aspect, but except for only one lithic insert, all the others were classified to have been cutting domesticated cereal. So it's confirmed that mostly these sickles were probably used for harvesting, cereal harvesting activities. We also made a further test with an experimentation, and Juan, me, and Fiona have been doing the past years, comparing the low high harvesting and the high harvesting. You can see the traces can appear pretty similar, but there is much striation and much abrasion in the low high harvesting. And so what we demonstrated here, there is some of the value that can clearly discriminate the high from the low harvesting in the experimental specimen. And here the results say that the discrimination canonical analysis demonstrate the archaeological tools are much more similar, the value, the discriminant value, to the experimental tools used for low height cutting. So this suggests that the sickles from Marmota, the 3 sickles that we studied at least, have been mostly used to cut near the ground, so to cut the entire plant to obtain and exploit not only the cereal but as well the straw. For what concerns the chemical analysis of the adhesive substance, this analysis has been done by Maxime Rejot, which has demonstrated the use mainly for these 3 sickles of pinacea resin. This is very interesting result, and it's also help us to design the experimentation. And finally, we have the result from the phytolith and pollen analysis that I will only briefly describe. The pollen analysis has been done by Daniela Roba and Rosanna Caramiello, while the phytolith analysis has been done from Marta Portillo. And we also studied the no pollen pollinomorphs that has been studied by Leonello Morandi. So I will just briefly present this result. And all this collaboration started with a collaboration with Daniela Roba, studying first of all the experimental sickles. So first of all, I tested with him this result for the pollen in the experimental sickle to see the signal that we can get from the experimental sickle, and to see how much the pollen analysis was similar to the result, to the condition of the field that I harvested. As you can see in the field, there were quite a lot of weeds because it was biological cultivation. And we found a lot indeed of pollen from not only from the weeds and from the cereal, but as well from the environment just around the field. And you can see there are value of the pollen quantities before the use and after the use. So you can see the difference, and there is much more pollen that is accumulated after the use because of the harvesting. Of course, a part of the pollen, especially for what concerns pine pollen, was already in the resin before the use. So we extracted from the archaeological samples very little quantities, micrograms of the resin. You can see here a little spot, and then we analyze and we get the result that we will describe more in detail in the publication. But what is interesting is that we have, of course, representation of cereal, both for probably hordeum and Phytico species, which is perfectly fit with information that we have from the carpological study of La Marmota that has been done and is ongoing by Mario Rotoli, between the others, especially from him. And you can see, very interesting, we have a very detailed information about all series of arable widths that are included into the field. And this is a very precise information because it's an information that we get directly from the cereal field. Of course, this signal is not the result of only one season. There is accumulated signal, pollen signal within the glue, because glue would more likely reuse and not completely erase every time. But this is probably is very interesting, and we will compare more in details with the carpological information. What is very interesting that we get as well some information about the type of adhesive. There is some pollen remains that suggest there is a Pinot-Halepsensis type. And then there is a lot of information coming from the fungal spores, but I will not discuss in detail in this presentation all these aspects for a matter of time. And as well, we get very coherent result from the phytology analysis. Then Marta can eventually answer to your question more in detail. But of course, we found woody taxa, including pine and oak, which is quite interesting. It could be both from the handle and both from the type of the resin. And then we also get the signals from the cereals, from the hordeum and triticum. So confirming the information we already have from the pollen and from the user analysis. So this preliminary conclusion, we can say that, of course, La Marmota is an exceptional site, and we are lucky to study these wooden tools. You have to imagine that me, Juan, and many other traceologists, we often look just to the little insert, and now we can study the entire tools, understand how it would use, associate a lot of residues to the lithic insert, and even understand some aspect of ergonomics related to the use of the tools. So you have to remember that these tools are one of the most emblematic tools of the Neolithic. They're a clear innovation. They do not exist before the arrival of the Neolithic. So it's a technology that has been imported into peninsular Italy and to the Mediterranean from elsewhere, from the Aegean and from the Near East, but with a lot of changes, with a lot of innovation that has been doing during this expansion of the Neolithic. So it's very interesting to study and to detail the rhythm of innovation that we have in our harvested technologies. These sickles are quite small, so between 36 and 24 of maximum length, including the handle. So the cutting edge is between 18 and 16 centimeters. This is quite similar to Caranovo, Anthrocycle from Bulgaria. And in general, it suggests that these tools were adapted to collect small quantity of cereal, because modern sickles, iron sickle, are even the smaller one. The cutting edge is more or less 30 up to 40, and then you have a larger tools. So the length of the cutting edge is directly associated to the quantity of grain of cereal that you want to harvest at each stroke. So one of the idea is that indeed these sickles were adapted to small scale agriculture and not to collect necessarily huge quantity of cereals. For what concerns the wooden material, these are not only selected specifically for the Sickle tools, because both Quercus and Rosacea and Malloidae species were broadly used alla marmotta. And for contrary, the use of pinaceous resin, I think, is one of the first attestation for the Neolithic, and so it broadened our perspective on the substance used by Neolithic people to do adhesive with birch bark and bitumen and other type of adhesives that are already known in the Mediterranean. So pinaceous is one of the first, probably the earliest attestation of use of the resin, at least for the Mediterranean Neolithic. And we can also detail how the system, the maintenance system of the tools that probably, as we already imagined from the studying of the lithic insert from other dry site, that probably it's mostly where this type of lithic insert, very small, were adapted to very short use with very little resharpening. Alla marmotta, there is almost no resharpening of the edge, but there is a use that probably we can quantify. We will try to quantify with the traces, but we can quantify to some dozen of hours. Then it's discarded. Not all the tools, probably just few of them are discarded and then replaced. One aspect that is interesting, I will just comment very briefly, that one of the Sickle, there is an abnormal quantity of pollen from Onenate pollen, and Daniele Aroa suggests that the recollection of this plant can be maybe related to the use of this plant not for alimentary reason, but because these species possess pharmacological properties, and when consumed fresh and in small quantities, it produces some alteration of the mental state similar to drunkenness. And you have to remember that alla marmotta there is opium, and there is the earliest opium of the Mediterranean with in situ domestication of opium. So maybe other plants were used for these properties. And in this case, we think that probably these sickles, which is a particular sickle because you have a decoration on it that the other sickle do not have, but this quantity of pollen is probably related to a last use of the sickle for the recollection of this type of plant. But by looking at the traces, we cannot affirm, and also looking the pollen information, that the sickle has been also used and mostly used for cereal harvesting. So this will be maybe saying the sickles, despite are being used mostly for cereal harvesting, they are as well used occasionally for collecting other plants, which indeed is very normal. But the interesting thing is that this plant may have been collected for other uses, and in this case, maybe for uses related to some type of industrial drunkenness or some other for pharmacological properties of the plant. So the next stage is to publish this result, and we hope to do it very quickly, and then to have a much more to improve our experimental frameworks to, for example, give an estimation of the exact duration, life duration of the lithic insert, and then we will also look for DNA, potential DNA trap in the resin, and including as well starch grain analysis, and of course do a much more extensive correlation between the pollen signal in the sickles and the pollen signal from the lake, from the site, and to relate all this information with the information we have about the ecological record and the exploitation of seeds that we have from the site. So thank you very much to you for the attention.
Thank you so much, Nicolo. This very holistic analysis of fascinating type of tool. Do you have any questions for Nicolo?
I would like to ask something about the state of preservation of the coast plain. And also, I saw like there was a photo where there was like a plant tissue on top of the surface. I just wanted to know whether there was a residue or like a root. That is the first question. And
I will ask there's a question about the state of preservation of the sickle blades, right? And there was one photograph that you showed that shows some residue on the surface.
Pointing towards like plant fiber.
Yeah, so you had an arrow pointing to something that looked like a plant fiber, and yes, and
yeah, maybe so. I can. There is some photos which are not directly from the Marmota sickle, but are just from experimental as well. So which maybe there I just use them. It depends what. If you, so of course the state of preservation is very good. I don't know if you are, if in this case, for example, I don't know if you are seeing, yeah, this plant fiber, this is just to show what we are looking to, but this is not La Marmota. So it's just to show it's not from La Marmota sickles. So this is an experimental one. It's just to show the idea of the study we have been doing. It's just for, so for what concerns the preservation is very good. Of course, in this case, we don't have the possibility to look at the microscope and the presence of residues directly on the lithic insert because they are too delicate, these tools, to be looked at under a microscope. So we mostly look with the impression to the micropolish, which is already a lot. And most of the plant residues are extracted from the adhesive, and this has been extracted by just with a metal tool by scraping a little bit the surface to obtain a few milligrams, and then these milligrams has been processed in different way because pollen and phytoliths they need different treatment to extract the residue in a good state of preservation. In all the cases that did not say that, but we have a lot of charcoals within the archaeological adhesive. So because probably charcoals, like a powder of charcoal, was added to the resin, to the pinnace resin, to create a more solid glue. This is very common as well in experimental archaeology. You can add to your glue ochre, need some abrasive elements. So the charcoal powder will probably use. So I hope I answered to the question.
And did you have another question?
There's someone else who wants to ask something, otherwise I have another question.
You can go.
I saw like another photo where you have like a cotton bud. I wanted to know whether you use any kind of solvent or something to clean before the analysis or not. Take out the solvent or something.
Yeah, so Emanuela is asking that she saw a photograph of a cotton bud, and she's asking whether you use some type of solvent prior
yes,
to
because mainly water, you need water to clean, not the tool, not the wooden, and not even the, you know, the adhesive, then, but just the lithic surface. You need to clean them, and you need to remove the Polaroid that is being used to the consolidator substance. That with water we can clean it, but of course this you have to repeat several times because otherwise in depression you get a lot of dirty materials. So we clean it, and we only touching the lithic insert and clean the surface with water and acetone when we were not able to have a clean impression. Indeed, we take several impressions, we check the impression, and if the impression was not clean, we take another impression, and it was mainly made by using some, as you see, some cotton, because it was the only way to work on the archaeological materials without manipulating the wooden and the adhesive. But we get, in some cases, we try to get not only superficial adhesive, but the inner part as well, so to avoid possible contamination from museum, you know, and the fact that the tools have been stayed for several years in a storeroom, and so in the surface you can have some contamination. But for the pollen phytoliths, we get quite coherent result. We also get quite a lot of materials Marta observed from the lake environment. So some diatome and some sponge spicula, which probably are post-depositional materials, of course, because of the deposition into the water. And this might affect some, of course, of the micro remains that we have on the surface.
Do you have any more questions for Nicola? Thank you.
There's a difference where you can distinguish between harvesting low on the probe and high on the probe. Why that is relates to
yeah, so well, I really hope I can replicate your question. So she's asking about the use well, and whether the different patterns, right, the high and the low patterns, as you say, might relate to silicification.
First question, why? How can you actually explain that you can see the difference?
Yeah, the difference.
Yeah. So her question is, first, how can you explain how you see the difference between the high and the low?
Mostly is because of, even if you don't cut in the ground, of course, there is much more soil particles in the lower part of the plant, and probably more soil particles are included into the process of polishing. And so that's why probably you have more striation, which is not a lot, but is enough to get a visual differentiation of the high and low cutting traces. And this visual pattern, different visual pattern, you can see from a quantitative point of view as well, you can identify. So yeah, probably it's just soil particles that are included in the plant. There is near the plants and in the movement, even if you are not touching the ground with a lithic insert, because you don't want to touch the ground, because you risk to lose your insert if you touch the ground. But it's just probably the particles that are on the plants in the lower part.
Okay. So thank you for your replies, Niccolo.
Thank you so much.
Are there any more questions? I don't really see anything in the online chat either. So I thank you, Niccolo, for your contribution. Thank you for joining us.
Thank you. Thank you for the question as well.
And yes, we'll see you soon. Thanks, Niccolo.
See you soon.
All right, so our next speaker is here. It's Andrea Tubanji and Emanuela Cristiani, Juan Jose Ibanez, and Avi Goffer, who's going to talk to us about Branson tools and dental calculus as means to investigate the use of wild plants in the Levant at the dawn of agriculture.
So first of all, I would like to thank Marta and Aroa for inviting me to present my presentation here to you today in this really nice environmental conference. And so today I will talk to you about my Mercury project we just started a few months ago, which deals with the investigation, the study of the exploitation of wild plants at the dawn of agriculture in the Levant, studied through groundstone tools and dental calculus. So we all know that actually the interaction between plants and humans dates back to the earliest phases of our history, and that played a major role in our evolutionary path. Plants-human interaction involved aspects related, of course, first of all, to subsistence, such as, for example, the gathering and consumption of wild fruits. But also it involved aspects related to craft and technology. So we have to think about the use of plant fibers for basketry activities. And finally, of course, plants and vegetables overall played an important role in the lifestyles of our ancestors. Let's think about the use of wood as fuel for fire. And to speak about this, we have loads and a really big plethora of archaeological evidence which testifies the importance of plants, especially in prehistory. For example, we could start thinking about the use of cordage by Neanderthals in a Middle Paleolithic site in France, which is testified by the residues that has been identified on stone tools. Also, another evidence is given by the pitted stones that have been found in the Australian site of Gesher Benot Yaakov in Israel, which have been associated as an indirect evidence of nut cracking activities. Again, also nice and important evidence are the wooden spears that come from the German site of Schöningen, which are dated to 300,000 years ago. Also, information come from bioarchaeological evidence, like, for example, the identification of caries on teeth of individuals coming from the site of Grotte Pigeon in Morocco, the place of the site, Grotte Pigeon in Morocco, which actually serve as an evidence of consumption of starchy food. And finally, there have been a really important one was the identification of plant micro debris, namely like starch granules and phytoliths, which have been identified in the entrapped in the dental calculus and also as well in the groundstone tool surface coming from several Mesolithic sites in the Balkan region, which we will hear about later.
Yes, I'm really sorry about this.
Okay.
I'm having technical problems with the online transmission and people can't see the screen.
Okay, great.
Yeah. So that's
Do we have time?
One second.
Okay. To continue, after showing you all these kind of evidence that we have in our hands, archaeological evidence that we have in our hands related to the exploitation of plants in archaeology, we have to focus on plant domestication, on plant domestication, which actually represents the climax of the human-plant liaison, and which happened around 500,000 years ago in the Near East, and actually represented a major turning point in our history. Indeed, plant domestication is a part of an overarching Eastern agricultural revolution that has led to considerable changes in behavioral, technological, and social terms, and which ended millions of years of hunting and gathering. Indeed, with plant domestication, humans actually changed their status and became from hunter-gatherers and fishers to food producers. And plant domestication actually led to significant transformations in the life of our ancestors, which included changes in settlement patterns, in the economy, and also in the relation between humans and their environment. So, plant domestication has been actually one of the most studied and debated topics in archaeology. Numerous, really high number of studies from different fields, which include, of course, archaeology, agronomy, archaeobotany, genetics, and geobotany, have provided multiple evidence, which have been mostly related to identifying the appearance of aspects such as aspects related to the appearance of morphologically domesticated plant species, and to identify which were and which are the specific traits that can be associated to domestication, like, for example, the loss of seed dispersal mechanisms, changes in grow habits of the plants, reduced seed dormancy, and actually increasing in the size of economically important organs. But as you can see in the slide here, there have been some aspects that have been not so well studied, or that have been little explored, like that I would like to call like unsolved questions. Like, for example, how did plant domestication transform the patterns, the established patterns of wild plant exploitation, and also how did plant domestication affected the technology, so the tools that are related to plant food gathering and processing, and also the dietary habits of our ancestors. So these are the questions which are at the base of the PATH project, which aims at investigating wild and domestic plant consumption by combining two different but at the same time related strands of evidence, which are material culture as groundstone tools and dental calculus. In this way, I hope to provide the population perspective on plant domestication. And also, one important aspect and novelty of the project is trying to apply the systematic application of confocal light microscopy in the study of usewear on groundstone tools. So, as I said, the two strands of evidence that will be studied in the project are groundstone tools and dental calculus. So, groundstone tools are useful mainly to investigate the activities related to plant food processing. Through the study of the function of the life histories of these tools, we can achieve information about the gestures that were performed in plant processing. Of course, the kind of plants that were processed, the stage of the worked material, and also the different kinds of phases that tools as groundstone tools may have been subject to during their life use. Also, the application of confocal light microscopy to the study of usewear on groundstone tools will allow, hopefully, to provide some methodological advances in the field of study of usewear. And it will allow us mainly to have information about the specific plant species that were processed, the intensity of use, and possibly some new and more detailed information about the activities that were performed. Along with groundstone tools, we will actually study dental calculus, which will allow us to reconstruct the ancient diet through the study of the plant microdebris, which remains entrapped in the calculus matrix. It will give us information also not only about the diet of our ancestors, but also about their health status and lifestyle, and also through the analysis and the retrieval of non-dietary debris, we can of occupational debris, we can reconstruct environmental conditions and also non-dietary habits. Which is, so going through the area which will be the subject of this research, we are in the Levant, and the sites that will be analyzed will span chronologically from the Natufian to the Middle Preparatory Neolithic B. So the sites are Einan, which is an early and late Natufian site of Netiv Agdud, which is a preparatory Neolithic A site. The site of Karajin in Jordan, which is the late preparatory Neolithic A and preparatory Neolithic B site, and Nahal Yarmout 38, which is mid-preparatory Neolithic B sites. So for the sake of this presentation, we will focus on the sites of the Einan, Netiv Agdud, and Nahal Yarmout. So the site, in brief, describing the sites that are being studied, the site of Einan or Ain Malaha is located in the Hula Valley in the northern Jordan Rift, and it has layers related to the Natufian occupation, which goes from 1,500,000 to 12,000 years before present. So it encompasses all the phases of the Natufian, from the early to the middle to the late Natufian, and is characterized by semi-subterranean curvilinear structures. The site of Netiv Agdud instead is located a little in the south in the lower Jordan Valley, and it's related to the early preparatory Neolithic A, and during the excavation there have been found numerous oval and circular structures. And finally, going southern, there is the site of Nahal Yarmout 38, which is located in central Israel, circa 34 kilometers from the coast. And it's a preparatory Neolithic B site, which is more or less dated to the 10th millennium BP. And it's characterized, it's a really small site, but it's characterized by a series of rectilinear structures with plastered floors. So all of the sites that have been selected for this this research, of course, allow us to study both dental calculus and groundstone tools. So the selection was also based on these criteria. At the base of our research and to study the function of groundstone tools is the design of a dedicated experimental framework, in particular the performance of sequential experiments. In our case, we are going to process vegetal materials which come from the study area with the modern replicas of the tools made of the same raw materials that we found at the site, which are limestone, sandstone, and basalt. And we will, for now, we are conducting the experiment which lasts 6 hours. And for each hour, we are taking casts of the surface, and we observe the surfaces under the microscope to actually gather information about the development of microware at each step, or to identify the possible specific patterns in micropolish, which are related to the plant species and the duration of use together. And also, of course, we are analyzing the modification of the residue appearance and their distribution through time of use. And more in detail, we are also interested in the morphological characteristics and mostly in the modification of the plant microdebris, like starch granules, mainly starch granules and phytoliths, which could change in relation of the use, the activities, and the time of use of the tool. So we are currently building a database and as detailed as possible reference collection of both user and the residue. Here you see a couple of examples. On the left, you'll see some experimental microware developed after 5 hours of use of the tool to pound and grind chickpea. On the right, you'll see another tool that was used for the same time for 5 hours to grind instead cereals. You can see visually the differences in the polish that are in the traces that are related to the two materials. And also, we are building, apart from the user collection, a residue reference collection. As you can see at the bottom of the slide, where you have some examples of starch grains from chickpea, from lentil, and from wheat on the far right. All of these information will be, of course, used to us to test the efficiency of the tools and to see which are the aspects related to the use of the tools, but also serve as of paramount importance because they serve us as a base on which we compare what we see on the archaeological tools. So both speaking about residues and user. So it's our main way of comparing and to actually provide reliable functional interpretation of what we see archaeologically. So as I told you, oh, sorry. Okay. No. Okay, I think we're good. Okay, so as I told you, an important part of the methodology applied in this research is played by the systematic application of confocal microscopy. And for now, we are actually looking at the experimental tools and sampling, as Nicola, in the same way as Nicola showed you in the previous presentation, the used surface to extract isoparameters, which allow us to distinguish, to discriminate between aspects of related to the working material and also on the time of use. So far, we have started, and we have just started, and we have analyzed only the tools which were used for the processing of chickpea and wheat. And as you can see from the scatter plot on the upper right corner, you see that we have a good amount of distinction between the working materials. So for now, we're happy with the results, and we hope that we will, of course, have more experiments and we will improve this, and then we will use it as a base to also include the evidence coming from the archaeological tools to see if they fit with our interpretation based on the qualitative observation of user. So coming to the archaeological tools that have been studied so far, as you can see from this slide, we have different kinds of tools. We have both passive elements and active elements. We have slabs, we have pebbles, we have pestles, and we have mortars, as the one on the lower left of the slide. So we are analyzing, we're not making a selection based on the typology of the tool. We are trying to look at all of the kind of tools which could have been employed in plant processing tasks. Coming for some of the results that we have to date, we were able to identify both residues and user on the tools that we analyzed so far, and we identified user related to the processing of legumes, as you can see in, I don't know if you can see the pointer, but you can see in the column on the left of the picture on the right. So you have on top the experimental wear, and at the bottom the user that has been identified on one tool coming from Enan Malaha. And you can see that the user is pretty similar. And on the right of the same picture, you have the experimental wear generated by the processing of cereals. And at the bottom, you have the user identified on one tool coming from the site of Netivak Dut. And you see how the traces look apparently really similar. Talking about residues, we have identified starch grains related both to the to cereals, to species of the Triticia tribe, and also starches which are related to legumes. One thing that we saw during our study so far was the fact that we identified a high number of starches showing recurrence damage patterns. So we were seeing this kind of forms, these starches that were damaged, as you can see on the left of the slide, and this pattern was recurring on several tools, especially from the site of Nahal Yermut. And at the same time, we knew that these starches might have been related to legumes, but we needed to have a proof because we didn't have the entire starch to say, okay, that's a legume. So what we did, we are currently actually proceeding in an experiment where we are trying to identify the damage patterns, the damage pattern which may affect starch grains, both of legumes and cereals. And as you can see here, on the picture on the right, these are experimental starches which have been processed and show really similar breakage patterns that what we see in the archaeological tools. So in this case, when we find this kind of residue on the groundstones, especially, we are fairly convinced, we are confident that these starches are related to the processing of legumes. And so going through the analysis of talking about what we see on dental calculus, and we are really happy because the residues that we identified, the starches in particular we identified in the dental calculus fit really well with what we see on the tools. So we don't have something completely different. So we have for now all the pieces that come together well. And we identified starches related both to cereals and legumes. So this pattern seems to come along. Of course, we have just started, so we need to enlarge our sample. We will go to sample again in this summer to have more tools and more calculus to analyze. Also, we need to complete our series of experiments, adding new and other kinds of plants, both from cereals and legumes, and also doing other kinds of activities out of the sphere of plant working, just to have like a comparison. And of course, we need to complete this first stage of dental calculus analysis, and also to conclude and to finish the analysis of residues and user on groundstone tools. So as I told you, this is a project that just started, and these are just the first results that we had. But I think that they show really well how different kinds of techniques can help us to investigate and to study actually one of the most debated aspects in archaeology, which is the advent of plant domestication in the Levant. So thank you very much.
Thank you so much, Andrea, for the talk. And, no, the problem is that we're connected. Yeah. Maybe we can use. I want to apologize for the technical.
Don't worry.
Come here to talk because no, not working. Mi prendo un sedio un attimo e dopo ti la devo fare. All right. All right, so that means that we can see the picture. Super, vamos a ver. One moment. Vamos a ver, el problema es la retransmisión por YouTube, ¿no? Porque es el micrófono del ordenador, eso es lo que me han dicho. O si hablo por ese micrófono también se oye por YouTube. Sí, sí. Vale. 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 Sí. Vale, vale, vale. 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 Vale, pues vamos a ver. This is So to apologize to Andrea for some technical problems, interruptions and stuff, and also to the people that are following us on the YouTube channel, because we are aware that the presentation is not in like full presentation mode. As I said, it should get better from now on, so our apologies for this. Do you have any questions for Andrea? Yes. Yes. Let's check if it works.
With regards to the radiate software, is it possible that it might be able to do like taxonomical regions and mechanically the microfossils and that is, or you think it's necessarily like a mechanical?
Well, we are pretty confident that it's something mechanical because the experimental starch, the damage that you see on the experimental starch are the starch that come from the groundstones that we use to work both the chickpeas and wheat. So this is one of the main reasons why we mostly related to the damage related to the mechanical stress that the granule may have been subject to during the use, more than like a taphonomical or post-depositional kind of alteration.
No sé yo. Se oye como raro. Sí, es lo que entonces, exacto. Are there any more questions?
Just wondering about your reference data set to be used where on the ground on the stone. If I catch it well, just been processing one type of plant per stone tool. And did you never combine them? Because I assume in the past they were not, purpose of the stone tool was not a single processing task. They make different things, the same object. So you should be able to discriminate or getting some confusion between the program.
Okay. So the question that I was asked for was that why for now we are just replicating the use on a single species of plants and we were not doing multiple uses on the same surfaces. This is a really interesting question, and this is something that we will do in the next step, because for now we are starting with it just to have the, let's call it pure or pristine user related to each plant. The next step of the experiment will be to do multiple uses, because of course we all know that groundstone tools are complex tools that may have been used for different kinds of activities and to work different kinds of materials. So yes, of course, this will be something that we will do in the next phase of experiments, and hopefully we'll be able to discriminate from a user perspective.
Any more questions? Okay. Thank you so much. All right, so the final speaker of this session is Leonello Morandi, who's going to be talking to us about the paleoecological proxies for prehistoric pastoralism, experimental data, and archaeological case studies from Liguria, northwestern Italy. Let's see if we can upload your presentation. Okay, this is all right. So I'm just gonna check very quickly if this is showing in YouTube. Marta, if you can check as well. Questo no è. Pionnello, tu che parli? Adesso perché si vede così? Non capisco perché. Questo è stato lo stesso problema che abbiamo avuto con Andrea, capito? Inverti, duplica, no. Ma io l'ho fatto. Sì, ho capito, ma non so cosa. Capito? Cioè io l'apro così, faccio qui, ma dopo si vede così. No. Possiamo provare, no. No. Nascondi, schermo. Juan, Matteo, we're having some issues with the presentation, the mode of presentation, and the way the PowerPoint is being seen online. So some people have complained, yeah. Se está compartiendo, pero claro, no se ve la pantalla completa. No, pues,
Compartir pantalla la primera. Ahora vamos aquí.
vale, y me das ahí.
¿Lo va a ver? Vamos a ver. Es que hay un problema que no se puede solucionar, no sabemos por qué.
Vale, vale. No,
Espera un segundito. No sé si... No se ve. No sabemos por qué se ha quedado así ahora. Es que Mateo ya nos ha dicho que no, antes le he preguntado, que no sabe por qué ha pasado. Tenemos que seguir así y ya está.
Okay, so yes, just as I was saying before, we're having some technical issues regarding the visualization mode of presentations. We know it's not ideal and it's not full screen. We can't really identify the problem, but we're going to be working on that during the post. So without further ado, I'm going to ask Leonella to say, and yes, I'm over here. Microphone for the YouTube channel is close to it. This is the microphone. This is the microphone for the room, and the microphone for the online transmission is the laptop. And another issue that they keep tell you guys is that there's and they need to exactly the chat because if you get too close to it, you're gonna fall. Yeah, somebody did it already.
Okay, thank you very much for inviting me here. So with this talk, I'm going to give you an overview about recent and current studies, paleoecological studies in Liguria, specifically focusing on topics like the significance of newly identified paleonomorphs, works on pastoral analogs from modern contexts, which could be applied to archaeological case studies, and the contribution of marine microshell assemblages. And hopefully I will also explain you something about the integration of this data with stable isotopes data. Liguria is located in northwestern Italy. It's a region that, although it has access toward the south to the sea, it actually has a landscape which is mainly characterized by hills and mountains. The archaeological evidence in the later prehistory of Liguria is characterized by both coastal and upland pastoral deposits with caves used as stables. And also there is evidence for anthropic use of the uplands as indicated by finding of artifacts which might derive from hunting activities, and as also indicated by a paleoecological record which points to deliberate forest burning by humans to clear up areas of the wood for hunting and pastoral purposes. Decline arises in tree taxa, which might be also influenced by human activities, and the accumulation of peat due to erosion, which has brought fine-grained sediment inside wetland archives. There are also between Liguria and Tuscany carved stones, which according to some hypotheses might indicate prehistoric Herdian roots the Copper Age shepherds in the region. So since about 10 years ago, the paleoenvironment of the plain of Genoa has been studied because Neolithic remains have been brought to light by the excavation of the underground, and new boreholes have been taken in the later years from the Piazza della Vittoria, which is on the alluvial plain formed by the river Bisagno, on which modern Genoa was built. And these boreholes have been particularly informative to get a much better view of the paleoenvironmental scenario in the area. So specifically, ecologically, these results are very interesting for showing us the presence of erosion indicators for brackish conditions in the plain of Genoa in antiquity, and also an important abundance of rotten wood and stagnant water pools, probably used for animals to drink, and the presence also of domestic animals in the area. And we might actually imagine something like this with Bisagno flowing in the plain, forming meanders, water pools in some places formed by oxbow lakes, and probably the ancient shepherds using the area to graze their flock. Now, more specifically, there are some very interesting microfossils found in the course, and some of them have not been identified to the genus, but only morphologically. However, these types have been recorded also in northwestern Europe, and they seem to always be associated with salt marsh conditions or some degree of salinity. And it's not really clear what they represent. There is, again, a breakage pattern which is always recurrent on them, which might be an operculum of some dinoflagellate-like microfossil, but there would be some genetics possibly to shed light on the topic. However, it is clear that also in this case, it's linked with brackish conditions in the plain. And other microfossils which are normally associated with Carex species can in reality be considered again as indicators of salinity in the area. So for example, fungal hypopodia with this specific shape, which are parasitic on other plants. There is one genus of them, and one species in particular, which grows in salt marshes, and which would fit very well in the rest of the archaeobotanical record for the area. So these types might be assigned to a specific genus in this case. And also another microfossil, which is normally just indicated by its number, by its code, can be identified specifically to a type of fungus which grows in an environment characterized by salinity. And there are very clear data on the mycological literature about this multicell type with this shape and this size, which is very likely exactly what we have in the core. As regards to grazing activities in the area, there is an abundant evidence for the presence of several types of coprophilous fungi growing on the dung of herbivores. And due to the more geomorphological location, we can easily rule out that there is a contribution by wild deers, ibex, or horses that weren't even in Italy at this time. So it is very likely that because also bones of domesticates were found during the excavation of the underground, we are looking at the results of domestic animals grazing in the area of Genoa, or the plain of Genoa, that was probably used as a pasture by the community settled in the area. And interestingly, also Pizzo wood was found in one of the boreholes, which some scholars think might represent a piece of a pile dwelling. So this would change very drastically the view we have of the occupation of the coasts in Liguria and the Neolithic. The thing is, the archaeological evidence is buried under 15-20 meters of alluvial sediment at this time. So it's impossible to carry out an excavation. What you can see is just a small patch of the scenario, and you can just analyze a few layers of that time through boreholes, or when they built the underground, there was a possibility to carry out small excavation. So this is a sketch of what the environment might have looked like with grazing animals. So moving on to works on modern analogs, there are some results I'll show you about the abundance of dung fungi in modern contexts of different types, like open air animal enclosures, roofed stables, dung paths, and dung droppings from almost 30 modern samples. And to talk about this, I need to briefly introduce you Sporophyllus fungi and what they are and their biology. So they are fungal organisms that colonize the dung of specifically herbivores, I mean, in particular herbivores. And they have very specific spores which can be easily recognized, and they are used as a proxy to detect the presence of herbivores in the environment. Now, it's not entirely clear ecologically what's the biological cycle of these organisms. It looks like once they are deposited around the fungus, if they get eaten by the herbivores from the grass, they need to go through the digestive system of the animal, where there are specific enzymes which would trigger the growth of the fungus, which would sporulate again, and the cycle will repeat. It's still not entirely clear, even to mycologists, if there is a clear association between the enzymes present in the stomach of the animals and the chances of life of the fungus. So the main thing to note about the abundance of fungi in modern contexts is that there are several cases where there are zero counts of dung fungi in different contexts, whether they are roofed stables or open corrals or dung heaps or individual droppings of sheep and goats. So this has relevance in terms of archaeology, because when you have zero counts on of dung fungal spores from an archaeological site, it doesn't necessarily mean that there were no animals present. So you always have to look for different dung proxies and looking at the ones which are better preserved in that specific environment. And the numbers never reach higher than 200 or 300,000 spores per gram. Now, interestingly, there are also some unidentified microfossils that even statistically plots very closely with other dung fungi, which are present in different places, both in contexts in Northern Europe and in Southern Europe, and they always appear in dung heaps. So they might be considered as new indicators for pastoralism. And I'm going to show you them in a little. So the main conclusions are that, as I said, spores are not necessarily present in dung samples. And importantly, the most dominant species are very often Sordarian thermomyces, whereas the literature about dung fungi in paleoecology until like about 10 years ago was dominated by records of Sporamiella. But you need to really look at the whole assemblage, otherwise there might be species growing there that are more frequent than the main ones. And importantly, higher concentrations seem to occur in contexts which are used only for some months a year, and not where animals are continuously sheltered, especially if they're sheltered there also overnight. It's possible that continuous trampling of the animals and compaction of the stable floor would impede the growth of the fungi, and so having a high density of animals counterintuitively would end up producing zero counts of spores or low counts, because the surface is too much disturbed by animal trampling, and they also need air to go through the sediments to be able to grow and sporulate, and this might be prevented by intensive animal trampling. So as I said, there are new paleonomorphs which can be considered as potential new dung indicators and grazing indicators, which were found in both in UK and in Southern Europe. So this must not be a coincidence. They need some type of dung-like substrates to grow, but they still don't have any clear biological identification. So they can just be described morphologically so far, but they might be new potential indicators of grazing pressure. And even more interestingly, one of the dominant taxa found in the modern samples consists of Thermometres stellatus, which is a very rare type of fungus, almost invisible in the mycological literature and almost absent also in the paleoecological literature, which need very specific ecological conditions to be able to grow and disperse the spores. So when you find it in archaeological sites, it would be very informative, although I'm not aware of any case where it was recorded, but it might just have not been recognized so far. So the species was even unknown to the biologists until the second half of the 20th century, almost absent in the literature, and it requires prolonged warm conditions at about 1560 degrees, and it's a colonizer of hay and dung. And it might actually indicate that there was a bacteria-driven warm heating up of the sediment, and there are in the literature cases of spontaneous combustion of hay triggered by the decomposition and the gases inside the dung. And you would actually not have this type of fungi growing in it if you had like a deliberate burning by humans, where temperatures would increase too abruptly, probably for this fungus to be able to grow. So it might be a further evidence to take into account in dung studies to differentiate the deliberate burning from some sort of spontaneous warming up of the hay heap. So to relate this with our archaeological case studies, we present you some results from the cave of the Renecandide, which is located in western Liguria. It's a very famous Neolithic deposit. Starts from the Paleolithic, but it has a very famous Neolithic phase, and it's characterized by the presence of Fumier-type sediments, which are scattered everywhere in Southern Europe, from the Iberian Peninsula to Greece and going through Italy. Which means an alternation, cyclical alternation of burnt, unburnt, dung, and ashes. There is a very large literature about it, so I will not spend too much time explaining it. And this type of sediments are present in the Renecandide. There is also a new section that was studied with alternation of burnt and unburnt layers. And what was noted is that, as regards the presence of dung fungi, for example, there is at some point a peak in one level of fungi which need animal dung to grow, and nothing in the other levels. So in terms of interpretation, based on the modern analogs, this might actually mean that during the formation of the sequence, resulting from a dung accumulation, burning of the sediments, new seedling episodes, burning again of the sediments as the sequence builds up. If the animals are taken out for some period, a period which we might estimate, because the coprophilous fungi, which are quicker to sporulate, need at least 3 days, and the slowest one need at least 3 weeks. So there must have been some sort of hiatus allowing the fungi to grow and to disperse the spores on the layers, which then will become trapped in the layer, and you will get a percentage peak in fungi. So it is perhaps possible that looking at these proxies, you might have an even better idea of animal management on the site, identifying episodes of where the stable was empty and the animals possibly carried somewhere else to graze or doing some form of transhumance activities. This will leave the surface sediments undisturbed for a period of time, which is enough for the fungi to be able to grow and release the spores on the soil. In the Renecandide, also fungi associated with particular plant was were found, and this is highly relevant because the pollen preservation from caves is very often very poor, and the pollen grains are squashed and broken and very often unrecognizable, whereas some types of fungi give you a certain association with edible plants which are not present in the pollen record, but that must have been on the site because they are exclusively associated with this type of fungi. There might also be evidence of truffle spores, and there is a paper about truffle consumption in prehistory from a study in the Pacific Ocean, but they might have been another food source. And also other parasites of plants like convolvulus, which were not present in the pollen diagram, but might be indicated, but are indicated by their associated fungi. And also aquatic species. Personally, I think aquatic species are a bit overlooked from stabling episodes, from stabling layers. They are highly relevant as they give us some indication of the environments where the animals were drinking, or from which the water was brought to the animals as a drinking source. In this case, we have algal cysts, probably from some sort of shallow stagnant water. Again, it's not very clear what one of these ones are. There is also a study about DNA extraction from it, but it wasn't like very conclusive about the results. I'll move on talking about a microarchaeological proxy, which is used very, very rarely. So as regards the plain of Albenga, which is one of the few plains present in Liguria, we have also started to study microshells from a core that was taken at the mouth of the river Centa. Now the ancient mouth of the river was located on in another location where you see the yellow symbol, and in the Middle Ages it shifted westwards. Now it is important also to note that there are Neolithic and Copper Age cave tables just 10-15 kilometers in the backcountry, and a suitable mountain pastures above it. So we're going to try to interpret this landscape in its entirety, and it is likely that people using the cave tables might have used the plain of Albenga for some sort of activities. So what the microshells can tell us is that, first of all, the assemblage is very diverse, very rich. Again, it is very deep, in like between 20 and 50 meters, the same problem we have in Genoa. So to get to the later prehistoric levels, because there was so much sediment accumulated by the river, it's almost impossible to reach that depth unless there are some boreholes, normally not carried out for archaeology but for other purposes. So there is a strong dominance of Lithium genus, Tricolli and Lucinella, and also levels with brackish indicators. So we really need to think about some sort of salt marsh environment, and also terrestrial species. So there were probably frequent episodes of marine regression and marine transgression in the area where the coast has shifted back and forth frequently in time, but mostly all these species are associated with seagrass, with Posidonia oceanica, which is nowadays completely absent from the beach assemblages, which means that in antiquity it is very likely that, because now this is the current distribution of prairies of seagrass in Liguria, and they are all at certain depths around 30, 40, even 50 meters, but Posidonia needs the light to go through, so it cannot grow any further ashore. But anyway, they're not so close to the coast in this place. So this is mostly probably due to modern pollution, but it also means that in prehistory, probably there was a high abundance of Posidonia on the shore, and which, by the way, it's not a seaweed, it's a proper flowering plant, but it grows under marine water. Current malacological assemblages are completely different, and we need to think about something, probably a situation like this for the Neolithic and the Copper Age in the plain of Albenga, where huge heaps of seagrass were available to the prehistoric populations. Now, this has some relevance. It is actually material which has several purposes in traditional societies. It can be used as a very good thatching material, and especially it's also used in stables. There are also modern stables where they collect it from the beaches and they use it as animal bedding. And there are also studies about the use of it as cattle feed, and cattle can eat it with no harm, and it's a good source of food for cattle. Now, macroshell assemblages give us also more specific indications about the ecosystem present in the area. So we found some parasitic species. Marcello Antoni carried out the analysis, who is a malacologist, and some of them are associated with specific animals, and they can only grow on these specific animals. Eulimidae are a family of microshells which are very small, almost translucent, and always parasitic on other animals. So by finding these microshells, we can confidently say that also some particular starfish was present in the area in the later prehistory, and another species of sea urchin was present in the area because they can only grow on this species. So these data really give a broader view on the whole ecosystem, not just about the shells that were growing there. Now, was there any interaction between the uplands and the plain in the later prehistory of the plain of Albenga, or was there any form of vertical transhumance? Now, we put together this with some very recent isotopic study. So, as I told you, there is a case table present in the backcountry that was used throughout the Neolithic and the Copper Age and part of the Bronze Age to stable animals. Was dug in the 1980s, and there was a very rich assemblage of sheep and cattle teeth. Unfortunately, most of them come from the upper jaw, and whereas most of the modern studies about the physiology of cattle and caprine teeth are based on mandibular teeth. So in order to have comparable data between upper and lower molars, we carried out some experimental work bearing a modern cattle skull, and the signals from the lower and the upper molars was compared, and they seem to overlap quite well. So the two signals are comparable. And what we found for the plain of Albenga in terms of possible grazing activities in the later prehistory is that actually the levels of delta oxygen are very low, much more negative than expected for a lowland condition on the plain. And also the content of nitrogen seemed to rule out any possible grazing on the coast and any intake of seagrass. So it looks like although there was a large availability of seagrass on the coast as evidenced by the microshell assemblages, they were not really taking the animals stabling in the uplands to graze on the coast. It is possible though that they collected seagrass for some other purposes. So how these conclusions were reached? They were reached looking at the oxygen 18, oxygen 16 ratios in modern weather stations from different altitudes, which are represented by the gray bars, whereas the horizontal rectangle represents the range of the Neolithic and Copper Age specimens. and even the higher values, they're still too negative to represent any intake from a lowlands coast. However, this is still not clear. We would need a larger study, including more caves and more zooarchaeological assemblages, also because the flat patterns of these records might actually point to some sort of seasonal vertical movement. It's difficult to interpret from an isotopical point of view, so more data would be needed. Thank you very much.
Thank you very much, Leonardo. Do you have any questions for him?
Mine was actually just but I have questions. I don't know almost anything about the downward Sickle method. I wanted to know whether that fungi or spores may also attack, like the ones that you have shown at the beginning, can also attack the human.
Because people, if you could repeat the question on that.
Okay. Okay. So there was a question about the fact if dung fungi might also grow on human feces, right? Okay. So only a few taxa grow on the feces of omnivores. Like more than 90% probably just colonize herbivore dung. But a few of them can grow on feces of wild boars, for example, pigs and animals. However, these species are quite rare. So when you find a large assemblage of dung fungi, it's possible that the contribution given by omnivores is very small compared to the other ones. What you would have to do to check the presence of human feces in the record might be to just look at intestinal parasite remains, which preserve very well. And they were also present in the plain of Genoa. So there was an abundance of parasite eggs from intestinal worms, which can come either from animals, from humans, sorry, or from pigs and boars, because they have the same size. Whereas cattle, sheep, deer, they have larger parasite eggs. But the ones which grow in humans and suids cannot be distinguished just morphologically. You would need to have DNA to do it.
Are there any more questions? I don't think congratulations to your talk.
Just like why if there's no one else, I just was curious about the asparagus. Like, was it like coming from the environment or were they eating something?
As far as I know, there is no recorded presence of asparagus in the cave. Now, where the fungus growing on the asparagus comes from, it might have been eaten like outside somewhere by the animals. It might result from plants that were inside the cave. This is difficult to tell because the taxonomy in cave is very complex, and there's also wind might have also brought it inside the cave. So we can say there were plants of asparagus in the surroundings. It's possible that maybe they were collected for as edible material, but the taxonomy of caves is too complex to give any certain answer to this.
I have a question for you regarding the experimental work that you've been doing. You mentioned the hypothesis that animal trampling might be preventing fungal growth.
Yeah.
Are you planning on taking, for example, microarchaeological samples to test this hypothesis? Or?
So far, I don't have any micromorphological sample taken from this context, because some are not sediments, but are just the dung droppings directly, which had not been trampled over by any animals. So the thing is, we have data from the shepherds, and we know how often they keep the animals there, and how often the site is clear. So we wouldn't probably need micromorphology to identify tramping episodes, as we already have interviews and the data from the shepherds, but it can also be tested experimentally if there's some correlation between zero counts and some lamination resulting from animal trampling.
Do we have any more questions? Okay, thank you very much, Pizzo. Interesting. We are 15 minutes ahead of schedule, so we can have a long coffee break. Okay, so we shall be back here at 4:30, so in about half an hour. I would thank the speakers again for your contributions, and I would kindly ask the speakers for the second half of today to approach the desk if possible to load their presentations. Thank you very much. Let's see you in half an hour. Que no, Marta, volvemos a ver siempre que el micro de YouTube es el del ordenador, que no es este. Ya. Vale.
Paste is
You can just
I copied. Where is paste? Okay, so yeah. Copiar? No. It's this one. Okay. Yeah, okay. It's easy to, yeah.
have you
No.
Okay, now it's ready to thank you very much. It shouldn't be. It's starting to. Okay, so really be. Yeah, it's everyone. It's everyone from the next session. So let's see. So I'm going to open mine. All right, so this is what I do. Vale. O sea, entonces yo voy antes que ella, ¿no? Vale, pues me lo dices y ya está, y me vengo, me subo y lo hago. A ver, que no sé.
poniendo y todo. Ah, bueno, no, ahora ha puesto él el... No, estamos viendo. El problema es que no podemos probar nada porque ahora...
No estamos.
Él se ha puesto, ha parado el YouTube ahora.
Ya, lo he visto. Vale. Es que alguien me decía por WhatsApp de si se pone abajo, que tiene una opción de modo lectura, que se ve más grande. No se vería pantalla completa, pero se vería más grande que el modo en el que lo estamos visualizando ahora.
Bueno, ahora vamos a tener que entrar otra vez, porque evidentemente estamos desconectados.
Sí. O sea, estamos conectados a internet, pero está...
Sí, estamos desconectados a internet.
Madre mía, tengo una sudada de la angustia.
No te angusties. Vale. Ahora estamos cerrados, pero teóricamente siempre.
Sí, claro, pantalla.
Compartir. ¿Qué lo tenemos aquí?
Podemos abrir, por ejemplo, que lo acaba de cargar Lucarini.
Así.
Sí.
Y lo que dice ella es
Modo...
visualiza tu otra diapositiva. No. ¿Sí?
Modo lectura. ¿Hay alguna parte que sea modo lectura por aquí? No, si es que todas estas opciones ya me las he visto yo. A ver, ¿esta? No entiendo por qué, no lo entiendo, pero si es que cuando probamos al inicio no pasaba esto.
Sí, no sé por qué, nos ha pasado alguna vez más, ¿eh? Y de pronto, pues, miras por qué. ¿Está saliendo? ¿Tú nos ves ahora? Sigue saliendo la sucesiva. Demostrar el lugar de la aplicación de impostor y digo eso dice. No. Primero de la esta es la imagen.
Es que, o sea, es como lo estamos viendo en modo presentador, o sea, como
sí, ya lo sé.
Ajá. A ver.
¿Ahora? Pero espérate. Espérate porque siempre tienes unos segundos más de retraso. Faccio una io, dopo tengo un café. Sí. La mano de Dio. El 10. Pues no entiendo por qué te dice aquí esto de compartir. No, eso se ha quedado ahí ese. Pero bueno, casi la solución es menor. Sí que se ve, creo, ¿no? La solución es mejor que la otra.
Esto lo he visto yo en ese.
Pero eso no lo puedes apagar, porque eso es el zoom. No, iba arriba. Y de aquí arriba. Cuando estaba aquí arriba, cuando estábamos con las dos páginas, se veía aquí y aquí. Sí. Había aquí un menú y había uno que ponía italiano como ver presentación o algo así. Pero no quiero salir porque si no la cagaríamos otra vez. Vale, pues,
Bueno, pues
vamos a ver.
sí, yo voy a utilizar el ordenador porque es que con el móvil me estás esperando.
Yo me voy a tomar un café, lo siento, pero
Sí,
es que
no,
estoy que me duermo.
sí. ¿De alguien?
Julio,
Este era tuyo. Son en Boston, ¿no? Cuba Libre o algo. Ah, sí, no me digas. No, para nada. No, pero será muy complejo, ¿eh? Porque esto de tener gente telemático es terrible. Además, hay un técnico abajo, es súper complicado. Yo ya lo he hecho y tal, pero no, claro, cuando empieza el trabajo, el aquí corriendo, pero tendríamos que haber hecho pruebas. Date cuenta, a ver, por ejemplo, presentamos una superguay, pero no sé, y también bueno, Andrea ha presentado también sus proyectos, pero no ha tenido resultados. Pero te digo que date cuenta que este micrófono es solo para la sala. Y que le digas que no es que vas a que le digas a la gente que no arrima mucho ya eso es lo más que repita las preguntas. Si solamente es este micro el que va. Bueno, van todos. Ya, o sea, todos están conectados a la sala, pero ya no te ven. Se va a YouTube, ¿no?
Perché la presentazione adesso farò un reclamo ufficiale.
Sì. Cosa?
Una persona...
Ah, ok, anche perché io mi muovo. Sono online già. No, sai come mi chiama? Con Morales, sai come mi chiama? A proposito di muoversi, come il mio soprannome, culo inquieto.
¿Pero tú estás viendo en YouTube ya, Julia? Entonces es que está en directo ya. Pero ¿por qué no se pone Julia aquí? La imagen aquí. ¿Por qué no sale aquí? ¿Sale Julio? Vale. No sé. Pues ya presenté. Okay. Espera un poquito con la presa, ¿no? Voy a hacer una foto de aquí.
Yeah, I have to be. Okay. So yeah.
Okay, so now we are already. Yeah. So 1 minute to go. Yeah. Welcome back to the Microarchaeology conference. Let's continue with our next speaker. Let's move to Africa. And our next speaker is Giulio Lucarini from the National Research Council of Italy and the Institute of Heritage Science and colleagues. So thank you very much, Giulio. Very welcome.
Thank you very much to the Spanish School of History and Archaeology in Rome, and in particular to Marta, Aroa, and Juan to organize this very interesting meeting, and also for inviting me to be part of it. So I'm going to present also on behalf of my two colleagues, Giuseppina Mutri from the Cyprus Institute and Anita Radini from the University of York. So what we are going to explore today is a bit the results that we obtained, we three colleagues. Yeah, it's okay. Oh yeah. probably. No, but here is active. So I really don't know, because here, yeah, it seems that is active. Okay. No, it's okay. So it's okay. Is it working? Okay. So different line of research that have been explored in the framework of different archaeological projects. The Mid-Africa project, which was hosted by the MacDonald Institute for Archaeological Research at University of Cambridge from 2017 to 2019. And then three field projects: the Farafra Oasis Prehistoric Project in Egypt, the Cyrenaican Prehistoric Project in Libya, and the Italo-Spanish Archaeological Mission at Melka, Kunture, and Balkit. So the main line of research were investigating sociocultural dynamics during the late Pleistocene and Holocene in North and East Africa, time of arrival and trajectories of dispersal of Southwest Asia domesticates into North Africa, pathways of low-level food production in North and East Africa, and how the Levantine domesticates were integrated actually with the African wild species during the Holocene, the so-called Neolithic. As a case study, I'm going to present today two case study more to the north in three completely different environments. One coastal, which is the cave of Awaftea, one desert environment, which is the Farafra Oasis, and then the last one where my colleague Giuseppina Mutri just started recently a new field project in Bifa cave in the highlands of Ethiopia. So when we look at the climatic sequence, basically we are talking about these two figures in the center. So after the Young Dryas, with the beginning of the Holocene, the shift of the intertropical conversion zone towards to the north basically allow climate to become more favorable. And so you can see from the presence of C14 dates that starting from actually the 9,000 roughly BP, the situation is pretty good until basically this limit, which is around 5,000 BP, when again the shift of this tropical rain belt to the south basically started this kind of deterioration of the climate in the Sahara, and then slowly the Sahara became the absolute desert that we know today. So let's start from the first case study. This is the amazing, wonderful Awaftea cave. This is a huge cave that opens right, you can see it just 1 kilometer from the coast. We are very close to Susa, the modern city of Susa, the ancient city of the Roman city of Apollonia. And the cave was excavated already during the 50s by a project of Charles McBurney, and then was reinvestigated from 2007 and 2015 by a project led by Graham Barker, again from the University of Cambridge. Reopening these very, very long sequence, we have a 15-meter stratigraphy covering basically the last 150,000 150,000 years of occupation. So basically from the Middle Paleolithic, Middle Stone Age up to basically nowadays, because the cave is still occupied by families of herders. In particular, today we are going to talk about two levels: the Neolithic sequence, which is roughly, you see the dates from 8.2 to 6,000 BP, and then these single elements coming, really exceptional element, a ground tool that come from the Upper Paleolithic, the Daban layer dated 31,000 BP. Speaking about the economy, before entering more into detail in the groundstone tool assemblage that we will present today, we can see very clear that even those, Holocene sequence and in these Neolithic layers, actually we don't have a big presence, of domestic fauna. Ammotragus lervia, Barbary sheep, unfortunately the labels are covered, is still basically the species best represented. And then we have a certain presence of domestic sheep and goat, and then presence, very, very low presence, and still not sure if it's domesticated or not, of cattle. What emerged clearly is the importance, even during the Holocene, even during the so-called Neolithic, presence of gastropods. I have to say that here we have marine gastropods, especially the species well represented is Forcus turbinatus. And then we have also some Elicidae and Trochoidea cretica and Rumina dequillata among the land snails. So this is a pattern which is very, very similar to what we can see in the Maghreb, so in the most western regions of Tunisia, Algeria, and Morocco. Looking at the plant macro remains, which were analyzed by my colleague and dear friend Jacob Morales, again, the pattern is pretty clear because even during the Neolithic, we have basically old wild species. In particular, in bold, I highlighted the species that are more present. We have Juniperus, we have Myrtus, we have Pistachio lentiscus, and as you can see, barley appears only starting from the Greek Roman layers. So let's pass to the other context, which is in a completely different environment. We are in the Eastern Sahara and very close to one of the so-called oases of the Egyptian Western Desert. So there are this series of oases that characterize the Eastern Sahara, basically west of the Nile, between the Nile and the border with between Libya and Egypt. As you can see here, you can see an image of here of a playa deposit. This playa deposit were some sort of lacustrine sediments on the shore of which semi-nomadic groups basically settled a little bit more during phases of more favorable climate and basically insisted in the territory. And the evidence that we have of these are these so-called slab structure sites, a sort of proto-villages that we can find in the Egyptian Western Desert, and that were basically particularly spread. You see the date, we have 8.6, 7.2 calibrated BP, 8.1, 7.5 calibrated BP. So during this time that correspond to the period in which this monsoonal belt, monsoonal rain actually was much more to the north, these type of sites clearly showing clear evidence of domestic activities are pretty much ubiquitous in the whole Egyptian Western Desert. What kind of evidence we found from these proto-villages? Again, mainly wild species. We have gazelle, we have ostrich, we have a massive exploitation, especially of ostrich eggshell. Emanuela here, I mean, a few years back in time, did really a very interesting study about our ostrich eggshell materials, and very few ostrich bones clearly pointing that these animals was exploited especially for the eggs and not for meat. And then very interestingly, we have among the first, is not the very first, but among the earliest evidence of domestic caprines imported coming from the Levant. We don't have in North African local domestication of cattle, of sheep, goat, pigs, wheat, and barley. They come from the Levant, all domestic, okay, already domesticated. If we look at the plant macro remains, is very interesting because, again, like in the coast along the littoral, we still have all wild plants, but we notice a difference. We notice the difference because here we have a clear, very clear presence of wild grasses. Above all, there is sorghum, wild sorghum, and then all other kind of species like Brachiaria, Echinocloa, Digitaria, Setaria, Urocloa. So all a series of wild grasses that are present here in the Sahara, but not so much along the African Mediterranean coast. So both the context, Awaftea and Farafra, yielded a quite interesting assemblage of ground tools. These are the ones coming from Awaftea. You can see on the top with the red background all the active elements coming from the Neolithic layers. And then bottom right, you can see these unique elements coming from the Daban layer dated 31,000 years ago. From Farafra, we have also an even richer assemblage. So here you have passive elements, the lower grinders. We have also in this case upper grinders, and it's very interesting to see that even in terms of numbers of these elements, we have much more in all the Saharan sites, much more than along the Mediterranean African coast. So it seems that these kind of tools were directly connected with the exploitation of wild grasses. In terms of raw materials, we have a different type of quartz arenite. We have a fossiliferous sandstone, so mainly all local materials. You can see here, you can see this kind of steinplätze, which are this kind of surface fireplaces that are directly connected also with this concentration of grinding tools. And also here you can see one of the slab structure site, in particular Eden Valley, and how it is connected along this kind of playa basin where this kind of lacustrine sediments should have been in place, and all around this purple triangle, as you can see, really a lot. Many of them are from the surface, so all the materials from the surface has been analyzing rather from a morphological, typological point of view, but few of them have been found from the excavation. And so for in case of that, those samples, we could try to investigate more in terms of use wear and also residue analysis, trying to understand better which was the function of these tools. The approach that we applied was a combined high and low power using a stereo microscope and a metallographic microscope. I have to say that this is a very lucky case. In fact, actually, this is not a grinding tool that come from Egypt, but from Morocco. So, I mean, you can understand also different regulation and rules. So Egypt doesn't allow the exportation even of environmental samples. So as you can imagine, it's really very, very difficult to work sometimes. But as also Nicola could show earlier, we could use Proville Novo. So we made basically custom molds of the areas of the surface. And so we could export at least the cast, the molds, and we could analyze them. Here on these a couple of slides that you see now, you can see this kind of example of microphotos. On top, you can see the ones taken with the stereo microscope, and the two down the ones taken with the metallographic microscope. So in terms of low power approach, so the analysis carried out with the stereo microscope, we could determine some kind of macro traces like edge rounding, leveled areas, some kind of grain extraction, grain fractures that can also tell us a lot about also the rejuvenation process of these tools that very often when the surface become very, very smooth, they could be repacked in order to keep the surface of the tool rough enough to be effective, because after using for a long time, the surface can become really very, very polished and very, very smooth, okay, and become absolutely ineffective. Then if you look at the higher magnification with the metallographic microscope, you can really appreciate the polish that develops on the surface of the single quartz grain. And then also in that case, using a reference collection, clearly try to understand, to have better ideas about contact materials, about the gestures, and all this kind of information that are connected to the human behavior. Okay, so here I want to report just a couple of examples. We actually detected this kind of spider web granular polish, which is quite reflective, which is the presence on high and low microtopography. It is very well linked and it mainly develops in patches. Okay, polish is always connected with striation, not always, sometimes connected with striation, and the directionality can change. It's quite random. And so this information is quite important because it gives us also information about the gesture and the movement. So actually we could find mainly this kind of evidence of rotatory kinetics. Then we, this kind of, according to a comparison with the reference collection, actually we could connect this kind of wear, this kind of polish with the exploitation of plants, okay? Then we have other kind of polish, which is even more reflective. It develops usually in individual grains, and also it shows a random directionality connected with striation, and this random directionality can change from spot to spot. And this kind of very high and very highly reflective polish has been connected with the stone-to-stone contact. So possibly even the contact between the upper and the lower elements. Actually, this is an example of one of these pebbles. As you can see, another thing that is pretty clear that in Farafra, these elements show a sort of technological traces. They were really manufactured, while in Awaftea, along the Mediterranean coast, we have mainly this kind of very opportunistic pebbles that were simply collected and taken. As you can see from one of these crevices, this one, very clear the presence of plant fibers that were stuck inside. And this is the study that has been done by Hugh Barton and Giuseppina Mutri about these beautiful upper grind from the Dabban layer. So we are speaking about a very old tool from the upper Paleolithic layer of the Awastea cave that shows this kind of quite developed polish associated again with striation and being also showing this kind of highly reflective, very bright spot that possibly has been associated with the processing of some sort of soft material. We didn't want to stop at this level of observation. We would like to go on. And so also we performed some kind of residue analysis, analyzing especially starch granules and phytoliths. And in terms of the methodology applied, we used also always the spot sampling, dry spot sampling or wet spot sampling using the pipette, as you can see. And here my colleague Anita Redini built really a very, very comprehensive reference collection with over 70 taxa, including, first of all the species that we basically retrieved among the macro remains in the different case studies, but also expanding these to other species, especially among the ones that have a very low visibility in the archaeobotanical visibility. So if we look at the starches and macro remains that come from these very old Dabban upper Paleolithic active element from Awastea, they found that these starches, starch granules that have been interpreted as belonging to the species Aegilops, this wild cereal that is still present in Cyrenaica and should have been present also during the Upper Paleolithic in the area. This is very important because actually we don't have so far any kind of information about the plant remains from these layers of the cave. From the Neolithic layers, actually we could determine only one type of starch granule. According to our reference collection, it has been interpreted as belonging to the subtribe Cencrine. I have to say that Anita and I always try to determine this kind of starch at the subtribe level, so we didn't feel confident to be deeper at the genus or even species level. So we are pretty confident to stay at this subtribe level. So from Awastea, only this kind of starch, and it's quite interesting because species belonging to the Cencrine were not identified among the macrobotanical remains. And much more interesting situation emerges instead in Farafra, in that area of the Eastern Sahara, where the presence of wild grasses is really very, very clear. And we could detect 5 types of starches. The first one was associated to the tribe Eragrostidae, subtribe Eleusine. It's very interesting because here we, as a reference collection, we consider the genus Eragrostis, which is not present among the macrobotanical remains. So again, this kind of information that complement in a way the information that we already got from the macro remains. Then we have another type of starch, which was associated to the subtribe Digitarine. And Actually, we have among the macro remains the presence of Digitaria sanguinalis, which is present, and so possibly we could associate this to that species. Again, we have another type of starch, which is larger, still polyhedral in shape, that has been linked to the tribe of Anthropogone, and Anthropogone possibly refer to the presence of sorghum, the wild sorghum that is present in really very, very strong number in the assemblage of the site. Another one, tribe Panice, subtribe Cencrine. So this again, this tribe Cencrine, that in this case we associated to the genus Pennisetum, and Pennisetum elatum is among the species present at the site. And the last one, again, the same tribe Panice, subtribe Cetarine. And here we have different species that belongs to this subtribe at the site. We have Brachiaria and we have Setaria verticillata, and we think, considering the size of the starch granules, that possibly this could refer to Setaria verticillata, the starch that we found on the ground tool. So in terms of presence of different taxa, the results obtained from Farafra, from the Eastern Sahara, are mainly consistent with the one from the macro remain assemblage. The microfossils complemented the results of the plant micro remains, because sometimes helped us to actually determine the presence of plants that we could not find among the macro remains. And in both the context, the Saharan one and the Mediterranean one, actually we can confirm the absence of the domestic crops. The situation is really very similar if we look at the rest of the Eastern Sahara. All the Holocene sites that have been investigated actually gave only presence of wild grasses, no domestic crops, not at all. So why going on the wild side even during the so-called Neolithic? Definitely wild grasses are one of the first, possibly most immediate responses to food shortages. They represent a low-cost and easily reversible strategy, which does not involve a great productive capacity. Relying on wild plants may be more convenient than cultivating in areas where there is a strong rain variation. And basically, these nomadic North African groups developed a more successful economy based on nomadic pastoralism that was mainly based on cattle along the Saharan region and on sheep and goat along the Mediterranean coast, integrated with the exploitation of wild plants. These are the same species that, and it's not a surprise, that are actually exploited even today in some Sahelian regions. And what about domestic crops? They are clearly limited to the two edges of North African regions. So we have on one side the Nile Delta, Lower Egypt, and on the other side we have Tingitana Peninsula, we have the Eastern Reef. They are dated between the 8th and 7th millennium BP, and the very, very interesting things that with the end of the Neolithic, basically to have a full farming package along North Africa, we have to wait the 1st millennium BC with the site of Altiburos, which is here. Okay, so incredibly, when the rest of the Mediterranean with the Bronze Age seems to explode in terms of relation, connectivity, North Africa become suddenly silent. So I used to call this a sort of unpacked Neolithic. of North Africa. This kind of adoption of domestic species is the result of a very slow process of acculturation and interaction between the local communities practicing a broad spectrum exploitation of the environment and groups of farmers from the Levant and the Iberian Peninsula, so coming in the Nile Delta and in Morocco. So developing different but very successful and low-risk economies based on hunting and foraging a number of wild resources, mainly fish along the Nile Valley, shellfish along Mediterranean coast, and plant along the Saharan regions, integrated with the domestic animal herding and farming activities limited to the two extremes. And after the introduction of domestic animals around 8.2 calibrated BP, these integrated and not replaced wild resources. So I would like to pass now, going to the quite quick second part of my talk, also to present another case study, which is the one of Bifa Cave, that belongs to the well-known Melka Kunture archaeological site, with the very well-known lower and middle Pleistocene record. Bifa Cave, it opens on the right bank of the Upper Awash. We are on the Ethiopian highlands. We are around 2,000 meters above the sea level. And as you can see, this is the very, very preliminary investigation carried out by Giuseppina Mutri, excavating this trench that actually exposed three different layers of occupation characterized by ashes and charcoals, and at the bottom with this kind of occupation floor with the square stones, burned clay, and lithic artifacts. Yeah. I'm sorry to so the data basically obtained are all this data you can see here is non-calibrated, so it's around 6,200 calibrated BP. You can see the very beautiful lithic complex of Bifa Cave, characterized mainly by the typical microlithic tools, especially lunates, which are made on obsidian, which are characteristics of the Ethiopian late Stone Age. And even in this case, preliminary evidence, a result of the use-wear analysis that possibly carried out on hand scrapers that can give us some hints about scraping and cleaning bones, so possibly associated to butchering activities. Even in this case, residue analysis, the starch granules, both of C3, so possibly Tritice, and C4, so possibly Panice plants, both of them present. And in terms of interpretation, we can possibly interpret Bifa as a temporary recurrent hunting camp with again, in this case, a broad spectrum exploitation of the environment. Last two slides, just to conclude and to see some kind of prospect for the future. You talk a lot, Andrea and Emanuela will talk a lot about the dental calculus. This has been a field that we didn't have the chance to explore so far, but we really would like, are keen to do it. And in order to determine not only information about paleodiet, but also on environmental situation, and also, and this is, with this I conclude, this really amazing project that Anita Radini just started about having hints about this kind of micro residues from dental calculus that can give us information about the craft activities and also health condition of these people. This taste of hard work actually is taking into consideration different case studies from late prehistory to Roman time, all from Africa. We have sites from Egypt, Libya, Sudan, and Tunisia. Thank you very much. I'm sorry to be a bit long.
Yes, yeah, we'll do the questions not in the by the end because just of the program and yes, I can see that there is something. I have to. Oh, sorry. Yeah. I don't know why it has come yours. I think it was open already. Okay. So where is yours? Yeah, it's open, the PDF. Okay. Oh, that's tricky because PDF, no. Would you want to? I mean, I don't know. Just try because maybe. Yeah, because until now I think it's
sorry, it's just like here.
Yeah.
No, here.
No. Yeah.
Where you were projecting something before?
Yeah,
here. Maybe here. No, it doesn't go. Maybe here. Here it is. Okay. And show it. Okay.
Yes, yeah, well, I just introduced you. Oh, you can, right here. Not too. I don't want to lose any speakers. Okay. No, not sure. No, it's fine. Yeah. Okay. Okay. Let's continue. The next speaker, well, now let's move to the Balkans. And the speaker is Emanuela Cristiani from the Università La Sapienza di Roma. I think everything is all right. Yeah, just thank you.
Thank you, Marta, and thanks also to all the organizers for this amazing opportunity to present some of the results of my project. That is not, of course, only mine, but of all the collaborators. And so I think we agree that for some time now, plants are no longer listed among the hidden foods in ancient forager societies. And there has been a lot of recognition of their use based both on ethnographic, archaeological evidence, and also many of the evidence come from the bioarchaeological field of research. So today, actually, I would like to, as I said, get this opportunity to share with you some of the results of my project that at the beginning was about hidden foods that are probably no longer should be called like that. And particularly, I want to focus on a case study, which is represented by Mesolithic foragers of the Central Balkans, that is this like red square that I showed here. And I want to discuss with you their familiarity with the plant foods like cereals. So my case study is the Dan Gorges, which is a region that is located in between the present-day Romania and Serbia. And in this region, there are open-air sites that began appearing in archaeological record with the start of the Holocene warming. And these sites are actually more than 20 and are located in river terraces near strong whirlpools. They actually, foragers were wise enough to use the landscape and the features, and especially the rapids of the Danube. And this sort of specific landscape facilitated the intense fishing and intense fishing operations. And in this territory, as I said, there are more than 20 sites that span a period of more than 4,000 years. And they go, as I said, from the Epipaleolithic to the Mesolithic and also to the early Neolithic occupation of the region, which is dated around 5,500 calibrated BC. So this region also is very, very important for people who are interested in the so-called Mesolithic-Neolithic transition. And in this region, one can observe all the sort of transformation processes by looking at the changes that affected this foragers community from a sort of diachronic perspective. As I said, there are several types of chronological occupation. And this significant transformation in the archaeological record in the region of the Danube Gorges happened in a very short time span of nearly 200 years, and from a period that goes from 6,150 to 5,950 years calibrated BC. And this period coincides with the emergence of early Neolithic farming community across the Balkans as a typical Neolithic communities. And there are a number of material evidence like stone and also ornamental tradition, but also data coming from bioarchaeological field like isotopic analysis and ancient DNA, which have provided very important data for understanding the specific scale at which this dynamic of transition happened and unfolded in this region. And based on these different evidence, for example, we know we have a very sort of clear idea that the contacts and exchange happened between early Neolithic farmers and the local foragers. And this involved also women, movement of women and material goods, and also this interaction sort of trigger the burst of cultural and symbolic creativity in the local farmers. And this creativity is mainly found in the so important and famous site of Lepenski Vir, which is well represented probably in, everyone who's interested in Mesolithic by the production of this exceptional boulder, like this that you see in the middle of the slide, which are sort of material culture reproducing hybrid entities between fish and human beings. So from the early Mesolithic onwards, several sites in this region, the region of the Danube Gorges, also became primary places for a continuous episodes of internment of the dead. And this allowed us to have like a data set, like of more than 500 individuals that were recovered at these several sites. And this mortuary record is actually really substantial. And through the analysis of this evidence, researchers have been able to sort of reconstruct also dietary habits of these foragers. And in particular, from stable isotope analysis and also from archaeological analysis, because like sites yielded really tons of bones, we know that the local foragers exploited area for, as I said, specialized fishing, and this happened throughout the Mesolithic and also during the Mesolithic and Neolithic transition. And these fish were migratory species, like, for example, sturgeons, but also they fished the carps and also catfish. And these, as I said, were primary food throughout the Mesolithic, and isotopes probably detected an intensification maybe towards the late Mesolithic at this site. It is important to stress that this food was not only, this animal, these species were not only, important species to eat, but for them were also important species to think about themselves. And in particular, they were good materials for the symbolic constructions of the personal identities of these local foragers. And we know already from 2014 that the cyprinid, this carp teeth, were used together with some marine gastropods as body and also dress decoration of the local foragers. And as I said, already in 2014, we carried out useware and residue analysis for reconstructing, based also on the location where they were found in burials, the way, the modalities that were used for dressing up these people. So based on, besides fishing, there are also evidence, and in particular based on the region, geographical proximity with the region of the Near East, and also the presence, for example, of plenty of antler technology that is associated based on useware to wood and soil working, but also the evidence, for example, of pollens of pollen in coprolites and patterns of paleopathology, but also tooth reduction, so wear on teeth. Some have claimed that maybe local foragers also gather plant foods, and this happened probably before the Neolithic transition in this area. But to prove that, we started within the project, we carried out a sort of pilot analysis, pilot study, and we work on dozens of individuals that provided, actually from the site of Blasets, the recently excavated site of Blasets, that provided the earliest evidence for deconsumption in this region of cereals, wild and domesticated cereals, already during the late Mesolithic and by 6,500 years ago, and before Christ. And these results come from the analysis, I said, of starch remains within the dental calculus, and probably are an evidence that sustains the existence of interaction and exchange between the local foragers and the immigrants, farmers, that is in a way already documented through other strands of evidence at the site. So further analyses were conducted within the project, within the Hinderfoot project, to try to expand this pilot data, combining two complementary evidence, a complementary line of evidence for Mesolithic plant food consumption, which comes from dental calculus, sending, of course, the data set, but also to the technology used for processing the plant food. And our archaeological sample this time was composed of around 61 individuals that were selected, this time from several sites along this part of the Danube, and also involving Neolithic individuals as well as a comparison. And this population in a way allowed for a very long-term diachronic perspective from the early Mesolithic, as I said, to the early Neolithic, and this sample was actually very heterogeneous because it included also males, females, and children, and also old adults. And to integrate the data coming from dental calculus, we also try to verify, the use of a new category of tools that appear during the late, actually since the early Mesolithic, but especially during the late Mesolithic, which is the groundstone technology or non-napstone technology. And in particular, the tools that are found abundantly at the site of Blasets, that you can see here in the map. So since the late Mesolithic at this site, but also in other sites of the Danube gorges, these groundstones are appeared, and we know that they are elsewhere associated with a number of activity, ranging from grinding, pounding, packing, polishing, both of different material that can be vegetal or mineral, even organic material. And we know that they are characterized by a very long functional life. At Blasets, we had already a sort of independent and indirect evidence for the use of these groundstones coming from the analysis of the the osseous technology that suggested, because of the evidence of the indirect percussion for technique to produce the blanks for points and other kinds of tools, the use of pebbles. Also ornaments in a way provided to us with the indirect evidence for the use of non-flake stone tools, because in particular in embroidery or related to embroidery activities for processing skin and tendons, because we found in another sample, we analyzed thousands of these sculptive ornaments, and we found like very well preserved residues of tendons that we know experimentally need to be sort of produced also using the non-net technology. So we have in a way an indirect evidence of this. So going to methods just quickly, dental calculus was decontaminated and extracted like micro debris in dental calculus in 2 different laboratories, and with the help of course of Anita Radini, we carried out in 2 different laboratories in order to also compare independently results. And we use generally optical microscopy, a cross-polarized transmitted light microscopy. For groundstone technology, things were kind of more complicated, because we had to analyze them at the Department of Archaeology at the University of Belgrade. And of course, as always happened, we had like Giulio knows very well, like the problem of storage places for these tools. And the workflow that you can see here in a way synthesizes the strict protocols that we follow to sort of control the modern contamination during the sampling and also the functional analysis. And for example, they were collected and stored in wooden and also boxes, and we had to sample residues from these boxes as a sort of comparative purposes. And already in the field, we created models using 3D close-range photogrammetry. And then these groundstone tools were analyzed at low magnification to sort of assess the state of preservation, but also to identify like residues already in the field for then sampling the tools. And we also use, of course, as here we have already specified several times, the high resolution silicon elastometer to later analyze the at high magnification the traces at the laboratory at Sapienza University. And here in the lab, we integrated both qualitative analysis, but also quantitative analysis by measuring changes in the feature of surface micro topography. And these data were integrated to the functional analysis, the qualitative observation that we did, and also to the residues that we found. So we, of course, use a very wide experimental reference, which involved several course, like plants collected locally for starch remains, but also a number of animal and mineral, for example, residues, just to have an idea how they appear microscopically. And also, as we had to carry out specific analysis on sort of wild series that are not very common in the wild, we had to plant them, we have to grow them, so to have enough material, like the edgelocks, for example, to process them for several hours. And then we use the local sandstone pebbles in several activities involving, of course, mineral and animal and different types of plant processing. So results, such granules were in dental calculus almost ubiquitous in all the individuals that we analyzed, and we often found them in association with parts of dental calculus. And we were very lucky to find like almost 6 morphotypes that were recognized. Mostly, as we see, are the starch granule belonging to the Tritice tribe, so 0. Then we have also aggregates that we ascribe to oats. We have several examples of the so-called forgotten millets. So we have Setaria or even possibly Andropogone species. We also have like reniform shaped starches that are associated with the legumes, and also some evidence for a triangular type of acorn that is like behind the logo, okay, but trust me, so associated to acorn. And we also found small granules that the morphology of which like suggested the assignment to berries, and in particular to cornelian cherry. And this was very interesting to us because recently, from mortuary context from the site of Lassos itself, we found in cremation graves cornelian cherry fruits that were certainly interpreted as remnants of possibly ritual eating or maybe drinking, even though we know that it's sort of difficult to interpret the sort of user goal of this species when found in grape. But again, the connection is quite striking. And overall, this like table show you that the most abundant microfossils, like the macro remain, are the starches from the tribe Tritidae that were found with more than 1,000 granules that were recovered in these individuals. And methodologically, we had to work a lot in order to characterize the distribution and the morphological variability of the Tritidae starch granule in ancient plants. And we did a very extensive experimental and statistical work on wild and domestic cereals from both like Balkan and also Italian peninsula. And we sampled around 11 species of Aegilope, of the genus Aegilope, also 2 species of Ordium, 2 of Triticum, and they were like grown locally. And so we selected around 200 starch granules that were like randomly selected and also measured for size and shape and also length measure. And the experimental starch granule of the Aegilope grain grasses show a more significant size distribution than domestic barley or wheat. And this evidence is also supported statistically by the statistic test we run, which highlights that the Aegilope starch measurements are significantly different from those obtained for the domestic species of Ordium and Triticum. And so our results indicate that the most of the starch identified in the archaeological sample from the Danube gorges is consistent with most of the species of the genus Aegilope, and that these grain grasses were actually very widely used and consumed since the early Mesolithic in this region. And then in addition to the starch evidence, we also collected 42 phytoliths, and we retrieved them in 24 individuals, and they attributed them to Poaceae grasses, and the multicellular characteristic structures of long cells were identified in some Mesolithic individuals, and the one that are the dendritic structures that are typical of the grass inflorescence were observed only in one Mesolithic individual, but they were actually abundant, as one can expect in the Neolithic individuals that we analyzed. And as you know very well, this type of phytoliths is very interesting to people working on plant food consumption, but also treatment, because their presence has been experimentally associated to mechanical destruction of husks and palms of cereals while grinding them. So we also conducted some metagenomic analysis on dental calculus, and I'm not reporting all the results, which are actually very exciting, especially for documenting the exchange, even in the flora, mouth flora, bacterial flora of foragers of the Balkan during the transition period with the Neolithic ones. But in this moment, it's interesting underline that this analysis allow us to detect the DNA molecules matching plants in 2 individuals, 2 late Mesolithic individuals from the site of Lassos, where we detected abundant reads mapping to the genome of Corylus avellana, the hazelnut, but also some mapping to the Betula pendula, the willow, that we know can be used, like the internal bark of the willow can be used because it's a very sweet taste. So in various processing, various food preparations, but also elderberry with the Sambucus nigra was detected with the DNA. And in addition to this, as I said, we analyzed also the microbiota of the Mesolithic foragers, and this allowed us to detect that these foragers possessed already in the early Mesolithic a higher functional activity associated with the breakdown of the dietary glycans, so carbohydrate active enzyme, which include, of course, also plant storage, the starch. So this was very interesting as independent evidence for the data we were collecting. So with regards to the results of the groundstone tool analysis that I conducted together with Andrea Tubanji here, we found diagnostic use-ware and residues on 44 tools, and out of these 16 were positively associated with the plant food processing, and in particular, based on, of course, experimental data, we can say that they were instant using for crushing and grinding rust grains, but also possibly hazelnut and acorns. And we also had a number, I think a number of also starches that were found, 121, that suggests the use also of trititrate, but also, again, starches of the berry, cornelian berry were found, and also possibly legumes were also found. We also run spatial analysis and surface texture analysis that supported quantitatively the results we obtained with the qualitative use-ware, so they were used actually for plant processing. So together with their use as plant processing tools, we also sort of supported those indirect evidence that we gathered from the analysis of osseous tools and ornaments, because we found evidence of the use of these stones also for skin and tendon processing, and we analyzed them also using biochemical staining that confirmed the presence of collagen fibers of different types, which of course, and together, as I said, to macro residues, but the biochemical staining just confirmed that our observation in a way were correct. So going to the conclusion, you've seen I have presented you with different strands of evidence which indicated that for almost 4 millennia, the foragers of the Danube gorges had sort of regular access to plant nutrients and also some sort of dependence of these plants that was able to sort of create a difference in the flora of their mouth. And there are these two complementary lines of evidence in a way support that these grains were also very abundantly used already in the early Holocene and already in the early Mesolithic, and so at least 9,500 calibrated BC. Of course, it remains unclear to what extent they were using these plants, but we can definitely say that even if we don't know how important these were in their diet, it seems clear that this practice in a way passed through generation in this regional context all the way to the contact with the Neolithic in the second half, the 7th millennium. And it passed enough and to develop a sort of familiarity which raised the need for creating a plant food processing technology for preparing what we describe as a coarse grain flour, which mainly was created using the tool in pounding gesture. And we suggest also the consumption of partially processed grains that is corroborated also from the recovery of grains really even stuck in the meloplast. They are not single in the dental deposit. And this actually is the difference that we found with the early Neolithic individuals that are generally associated with the retrieval of single grains that also show a very developed like damage pattern, which we don't find in the granules that are associated with the Mesolithic people. And this probably pattern is associated with a very long process of grinding them for obtaining the fine flour. So based on this evidence, at least what we observe on archaeological tools and teeth, we suggest the probable existence of two different grind processing techniques associated, one with the local forages and probably another one associated with the Neolithic ones. So it is possible that this long-lasting interaction with these edible grains, but also the pulses that we found as starch that documented in this region from at the end of the late Pleistocene, might have allowed enough time to develop a sort of specific eating habit, what is in anthropology called like the cultural valuation, the palatability of people for specific tastes, and also such a shared knowledge about specific plant resources in this region predating the introduction of agriculture. Agriculture might have even, we can say, eased in a way the introduction of domesticated species, as we found already at the end of the late Mesolithic from Neolithic foragers. So we in a way found an ulterior evidence of this contact studying the dental calculus and the stone tool. There was a last slide. Well, I wanted to thank all the, but it's black, I don't know why. There were all the contributors, and anyway, thank you very much. Sorry, I was afraid I was not in time. I was a bit running. I hope everything is clear, but anyway, I'm here to answer all questions if you have any. Right, here, I was one. Hello. Oh yeah, later. Yeah. Okay. I just can stay here until 6:30. Yeah. Sorry.
I hear a lot about being related to food. What about the Cornelian cherry?
Well, sorry, I interrupt you. There was the. Shall we repeat for the people? So the question was about the use of Cornelian cherry, or I guess the way, I suppose this remains, are related, kind of processing was possibly suggested for this recovery of Cornelian cherry starches. Actually, I was also surprised. But then because okay, of course, seeds are found, and for example, in this cremation pit, they are found seeds, and also from the excavation, the Cornelian cherry seed is one of the. Sorry. So the seed was there, so and that's one evidence. But I believe that these starches that we found on the stones are related to the processing of the rest of the fruit, and there is an ethnographic also documentation of the use of berry, and also the use of berry together with fish, and also together with meat, to sort of creating a sort of probably different taste. There is from North America, there is plenty of evidence for different types of berries to be used. So I guess that might be one possibility, that they were grinding to sort of creating maybe a dry fruit, then to be turned into a flour. And I was suspicious about that, but then I found ethnographic evidence. So it might be also interesting to maybe try to expand on this sort of with experiments that also connects to, of course, the different use of the same tool with different material that might be vegetable or animal.
But people also oil or soap stones. You know better than I'm working on precisely in this context, but that could also be touching on the
Oil.
The hard part, the inner part.
Oh. Yeah, it could, yeah, maybe because you think it's the oil that you can gain from.
So that was the reason to ask, like,
Yeah. We found the same more abundantly in the seeds. This sort of starch were present and more in the seeds, but I don't know why I automatically thought it was from the fruit, but maybe you might be right. They might be a process, both. Yeah, 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 yeah. That's actually a good observation. Yeah, maybe. Okay. Thank you.
No, ahora toca, no cambiamos, ahora toca Aroa. Sí. Falta de Pizzo, que lo va a hacer. Vale, a ver si está. Está bien, ¿no?
Sí, no, pero va a pasar lo mismo que antes.
No.
Está igual que allí.
Sí.
Sí, ¿no? ¿Qué? Hemos hecho antes esta vida.
¿Te acuerdas cómo hemos hecho antes? Si te digo la verdad, no sé. Hemos hecho con la ventana, o sea, con la compartir pantalla. Desde compartir pantalla, o sea, algo que has hecho desde ahí, en el sentido de qué pantalla compartir.
No sé, no. Pues no sé. Es que esto, no sabemos por qué hace este tipo de cosas. F10 dices.
Sí, y ahí se ven opciones de pantalla, de división de pantalla.
Duplicado, ampliar,
Eso
solo segunda pantalla.
Eso es, dale a duplicado. Dale a duplicado. A ver si ahí os sale la pantalla y ahí. ¿Vale? No. Sí. Bueno, pues dale otra vez a F10 y ya está. Vamos, que le das a salir.
No, pero no sale.
Sí, pues dale con el cursor, como lo tenías antes. Es que yo pensaba que con eso se iba a seleccionar. Ahí. F10. Entonces ahí os salen unas diferentes.
No, pero ahora tengo que compartir pantalla por huevos, vale. Que no la tenemos compartida. F10 por duplicado, no sé.
Pues por ir probando ahí. O segunda pantalla, o sea, ahí.
No, segunda pantalla es lo que hemos hecho antes.
Vale, pues entonces, a ver, en esas opciones de F10 yo es lo único que...
No, pero tú sigues estando aquí.
A ver, sí, pero a mí me da igual.
Claro.
No, pero...
Claro, tiene que salir ahí. Mira, ahora está haciendo algo.
No está haciendo nada.
Sí.
Estaba listo. No, creo que está con la miniatura. No sé. No.
Mateo no anda por aquí, ¿no? Sí, pero bueno. A ver, Mateo. Yo creo que es una cuestión de compartir pantalla. Pantalla de... No, de PowerPoint. Hombre, lo puedo pasar.
Vas a tardar más que dejándolo así. Que no es un problema.
Sí, no pasa nada. Porque se vea pequeño, pero visualización. No quiero esto. Quiero esto. Estoy compartiendo.
¿Está bien? No, sí, están igual. No, volver porque ella ha hecho varios intentos. Yo lo dejaría así, ¿eh? No,
Sí,
que no se ve mal del todo, tampoco así, tampoco. Eso es la pantalla, de lateral.
Sí, aquí se sale entero, ¿no? Pues no.
Ahora es posible que esté bien. Ahora es posible que esté bien. Quiero estar viendo. Esto es súper directo, ¿no? Ahora es posible que esté bien, ¿no?
¿En serio?
Claro, ahora tienes el chat ahí.
Increíble. Espérate a ver. ¿Todo bien? So, I'm here because there has been a change in the program, so I'm going to go before Alessandra Pecci, who's scheduled to speak now, but she's going to be the last speaker of the day. So, is it okay, Marta?
Mira, ahora lo que se está viendo es que llegamos a un diferencia que pero ahora se está viendo así. No sé cómo está para...
¿Y cómo está
No, ahora se ve mejor. Claro, sí, es así. Es que ahora no está, ¿no? Sí, he escogido una cosa.
All right, so apologies for the slightly delayed start. In this talk, I'm going to share with you the scope of my Marie Curie postdoctoral project, which I'm carrying out at the Milà Fontanals Institution in Barcelona, and it's focused on the integrated geoarchaeological analysis of settlement open spaces at Neolithic sites from Turkey, Jordan, and Syria. I only started this project about 3 months ago, so unfortunately my available results are quite limited at the moment, but I have some data already from my Turkish sites from previous positions. Hence, this talk is going to be mainly a statement of intentions for the next 3 years for me. Okay. So approximately 12,000 years ago, mobile human groups began to develop a sedentary lifestyle supported by an expansion of mud brick architecture. At this time, new paradigms in social organization began to emerge, eventually resulting in the development of communal versus domestic spheres and concept of space. It would not be accurate, however, to assume that all settlement open areas of the early Holocene equal public spaces and that Neolithic buildings were always used for domestic purposes. In Southeast Asia, we have examples of communal structures, such as the central subterranean building of Jarf el Ahmar, interpreted as a communal food storage facility, or the ceremonial rooms or so-called temples displaying terrazzo floors at Nevali Chori. Equally, there are also several examples of communal open areas, such as the central activity area surrounded by large building compounds with no street systems at Hajalar, or the large open areas at built-in residential quarters at Chayonu. In spite of the ubiquity of settlement open areas that we see in many Neolithic sites, scholarly attention has long been focused on built environments, partly due to the high degree of standardization and symbolic complexity. This elaborate material symbolism led to the development of the concept of household identities, where the house becomes the integrative element of the community, overriding other kin affiliations. The appearance of households as transcendental institutions has centered the attention on buildings, and as a consequence, Neolithic research has long privileged household-level perspectives with large areas of settlements, that is, open spaces and open areas, often not being considered or subjected to investigation, at least not to the same degree as buildings. So the question that arises then is, what constitutes a settlement open area? In this research, we consider any space that's not covered by a fixed architectural feature, such as a roof or other permanent covering, an open area, regardless of whether walls are present or not. This classification includes passageways, middens, and spaces between buildings, the rooftops of cluster settlements, and abandoned structures without or with flat roofing. Now, we do realize that this definition is, of course, very broad, and it's quite open to debate. So this is not meant to be written on stone. We're just taking this as a starting point for this research. Thing is, the heterogeneity of Neolithic open spaces, we have defined them, poses quite a few challenges to their study. Conceptually, they represent transitional zones between the inside or the house and the outside or the landscape. However, the lack of well-defined physical borders in many settlements complicates the association of open spaces with specific social units of analysis in many settlements. At times, they are viewed as an extension of the domestic sphere, such as with courtyards, and at others as the passive containers of accumulated refuse and waste where no activities were taking place. And other times, they're seen as the scenario of super household interactions or communal activities. One of the main difficulties associated with study of Neolithic open areas is that they often display very complex stratigraphic histories, particularly in Neolithic sites in the Near East, an aspect that has also contributed to making these spaces overlooked. In many cases, they are constituted by very finely laminated stratigraphic sequences with layers measuring about 1 millimeter in thickness and represented single depositional events. Thing is that more often than not, these spaces are excavated in arbitrary units that do not always represent units of deposition. Now, this is very problematic because it creates artificial palimpsests that also affects the fines and environmental materials that are collected from these areas. In addition, the impact of post-depositional alterations such as bioturbation and salt reprecipitation in the form of gypsum, for example, tends to be much greater in settlement open areas than in building interiors, which is an aspect that adds to the perceived homogeneity of these sequences in some settlements, and ultimately to their stratigraphic complexity. So the main goal of the Patios project, which is my Marie Curie research, is to offer a new perspective on the socioeconomic trajectory of early settled communities in the Near East by attempting a shift away from building level analysis due to the complexity of Neolithic open areas and to the great amount of information that they contain on a wide range of activities representing ecological strategies and choices and daily practices. Their study is bound to be multifaceted. So we have divided this investigation into several interrelated research themes and sub-themes that represent the backbone of this project. The 4 blocks into which this research has been divided include formation processes, plant and animal management strategies, spheres of interaction and activities, and taphonomic considerations. The coalescence of these multiple facets of this research will result in the production of a novel body of work that will challenge current perspectives of community ethos and lead to a better understanding of sedentary practices. So the life histories of settlement open spaces will be reconstructed through an interdisciplinary multi-scholar methodology that combines geoarchaeological, bioarchaeological, and ethnographic techniques. The integration of this method will result in a more holistic understanding of open areas and their socioeconomic significance and evolution during the Near Eastern Neolithic. And we hope that it will provide a methodological blueprint for the investigation of open spaces and at other relevant settlements, not in other regions and periods. So this research focuses on 2 highly significant regions for the transition from mobile hunter-gatherers to sedentary communities, which is the Levant and Anatolia. These areas each constitute a biodiversity hotspot and a major regional hub for the transmission of economic, technological, and cultural innovations across the Near East and into Europe between 8700 and 1600 calibrated BC. The sites selected for analysis are unique for the representativeness of diachronic human settlement changes and ecological strategies during the early Holocene in Southwest Asia. These are Holocene in Jordan, Tel Karasa in Syria, and Bonjuklu and Çatalhöyük in Anatolia. Due to the very early stage of this project, results from Holocene and Tel Karasa are not available yet. So we're going to present some of the provisional results that we have so far from our Turkish case studies. So starting with Bonjuklu, it's a Neolithic site in the Konya Plain of South Central Anatolia. It's approximately 9.5 kilometers north of Çatalhöyük, and it stands above about 2 meters above current plain level, and it covers an area of approximately 1 hectare. The results presented here comprise samples collected from Area M, which is situated in the very center of the settlement, and it consists of mainly open-air medium deposits and short-lived built environments. So all domestic buildings at Bonjuklu are situated around this open central area in clusters. They consist of an oval structure demarcated by mud brick walls with occupation surfaces below their contemporary ground level, which display a major division in floor area between clean areas that are plastered with calcareous materials and sunken dirty areas consisting of ashy trampled surfaces, which is a very similar concept to what we see in houses at the later site of Çatalhöyük. So visualization models of the use of space for sleeping in a standard domestic structure at this site suggest that buildings could accommodate 2 to 5 adults in a similar number of children. The same models also indicate that these buildings could host larger groups when engaged in social interactions indoors, although space for inter-household activities inside houses appears to have been limited. Therefore, we need to look at open spaces if we want to examine activities beyond household level. Non-domestic structures such as the monumental buildings at Göbekli Tepe or Çayönü or Aşıklı Höyük, which were designed for the interaction of people beyond household level, are not really present at Boncuklu, and neither they are at Çayönü Höyük. Interestingly, there is a significant amount of evidence for the performance of ritual practices in open spaces at Boncuklu. This consists of decorated stones, plaster vessels, and a number of articulated burials that have been found in Midden contexts, indicating the important role played by open areas in ritual activities, in addition to food production and craft activities. Further, we also have non-standard constructions that are they consist of light superstructures, thin or temporary walls or boundaries perhaps, and variable floor configurations that also occur in this open central area. These have been referred by the team as non-standard buildings, and they have silty floors, no signs of prepared plaster surfaces. And they consist also of a variety of frequently repositioned features that include temporary hearths, plaster basins, and shallow pits. So the apparent dynamic nature of these structures points to the possibility that they were communal constructions used by several households for storage, cooking, or industrial activities. And this is an aspect that we're going to test by looking at the microstratigraphy of these structures. The stratigraphic sequences found in open areas of Boncuklu that we have had the chance to study so far really point to an anthropogenic origin. Organic inclusions, mainly char plants, bone fragments, and shell remains account for approximately 50 to 70% of these external deposits. However, although middens have a remarkably similar composition throughout the settlement, microstructural differences are clearly distinguishable between in situ activity areas and damped deposits. So we have multiple evidence for in situ burning activities that has been identified in Area M as manifested by the several temporary fire spots and more formal fire locations with plain line boundaries that have been unveiled during excavation. Local plant resources, specifically reeds and grasses. This site is situated in a wetland, kind of marshy area, and they appear to have used these local plant resources for fuel, the preferred source of fuel, which has been suggested by the phytolith assemblage. Quite interestingly, small aggregates of suspected herbivore fecal matter have been identified in a couple of these open fire spots. This evidence has been used to suggest that the collection and use of dung as fuel was taking place at Boncuklu, which is quite crucial because it confirmed this is a behavior that could possibly predate the domestication of caprines in this region. I have to stress, though, that this is still preliminary. The amount of fecal matter that we have found is quite sparse, so it raises questions as to the intentionality of dung use as fuel. And again, this is one of the research issues that the Patios project is aiming to clarify. Herbivore penning areas have actually not been identified in the Boncuklu sample so far, but when I first started working at the site a few years ago, I was trying to find evidence of pig or wild boar penning, since this is the most abundant mammal species that is present in the Archaeological assemblage. We thought we had it, when we found this buildup of fecal materials in the southwestern corner of the archaeological trench in area M. These fecal aggregates that you can see in the slide, they consist of up to 80% digested amphibian and bird bones, but they are devoid of trampled indicators which exclude the possibility of a stabling location. In fact, we carried out biomolecular analysis at the National Environmental Research Council facilities in Bristol in collaboration with Dr. Ian Bull, and these have revealed very high content of copper standard and cholesterol for these fecal materials, which point to their human origins. Therefore, this space has been interpreted as a latrine, and even ever since our identification, these human coprolites have been consistently collected at the site for further analysis, including parasites, DNA, proteins, etc. So we do hope that in the years to come we will be able to shed important light into the life conditions and health of the first sedentary communities that we found at Bonjuklu. Our second case study needs little introduction. It's the Neolithic megasite of Çatalhöyük, which is a vast till site that covers more than 13 hectares, spanning more than 1,000 years of occupation. We're moving into 30 years of excavations at Çatalhöyük, and these have revealed a Neolithic occupation defined by mud brick buildings constructed atop one another and at intervals separated by open refuse areas referred to as middens that are tightly enclosed between densely packed buildings. At times, and in the north area of excavation specifically, we appear to distinguish axes of open spaces that define groupings of houses, as Ian Hodder has referred to as neighborhood within the settlement. These open areas contain hidden deposits, and the assumption is that they served as conduits for movement within the site. Again, we're going to test this hypothesis with the samples that we have been collecting. Because it may be what becomes apparent is that open spaces occupied large areas of the Çatalhöyük settlement in spite of its very clustered nature. The site consists of two mounds, the Neolithic East Mound and the Chalcolithic West Mound, and of several excavation areas in both. In this project, we investigate the middle and late occupation levels of Çatalhöyük in the north area of the East Mound, and crucially, the latest levels of occupation, which have been excavated by Polish teams in the GDN area and the TPC area. This is particularly exciting because in these late Neolithic levels, we see a trend towards increasing house autonomy, which is likely related to changes in social organization. So in our project, we're going to explore how this process affected the layout, use, and accessibility of late Neolithic open areas, and whether we can detect the occurrence of domestic courtyards and the emergence of discernible differences between household and community spheres. In reality, open spaces at this megasite have begun to attract research interest in recent years, and we have recently published some of our findings in the project monographs that are slowly coming out of the press. We'll see. This chapter in particular represents a massive effort to bring together material analysis and environmental evidence and combine this with the stratigraphic evidence. However, I have to stress that some of the limitations of our work are that it relies quite heavily on the stratigraphic record of the site as defined during excavation, which in the very great majority of cases was conducted in arbitrary units, and crucially, it leaves out an important type of open area in clustered settlements, and at Çatalhöyük in particular, which is rooftops. So the discovery of roofing materials in space 488, which is in the central part of the north area of Çatalhöyük, allow us to mend this and to gather new data on rooftop activities at the site. Roof contexts are only very rarely encountered and securely identified during excavations. hence the importance of this find. In most cases, only the structural elements are present at the top of walls, such as large mud brick slabs for stabilization, or the wood beams, particularly in burned buildings. Sequences of plaster surfaces are extremely rare, but their importance stems from the fact that they allow us to investigate what was going on in the uppermost part of buildings, including the presence of upper stories. In addition to rooftops, this project will explore the so-called streets of Çatalhöyük. These are the linear open spaces identified, apparently dividing houses into sectors where meat and refuse accumulated. We will be looking particularly for indicators of heavy trampling, pointing to the use of these areas as passageways to facilitate movement of people and animals within the settlement. So I'm going to finish this talk by presenting briefly our previous research on pending locations at the site. We identified several herbivore pens at Çatalhöyük through micromorphology, although all of these belong to the earliest occupation phase of the site, such as a sequence in space 620 that you can see in this slide. And these locations precede the expansion of the settlement that we see in later periods. So ethnographic research in modern Turkey has shown that penning deposits are composed of phytoliths, ferrolites, and organic matter, often displaying ferruginous staining caused by the decay of organic components. Distinguishing between sheep and cattle dung in these modern samples is relatively straightforward, with sheep dung occurring in pellets and cattle dung displaying a micro-layered appearance. However, in archaeological samples, this distinction is far from easy, as both types of dung suffer from multiple modifications, particularly in penning locations, which include trampling, reworking, burning, leaching, just to mention some. At Çatalhöyük, pen deposits are characterized by multiple superimposed trample layers of herbivore dung, rich in phytoliths, and displaying iron staining together with abundant mineral grains, sediment aggregates, and charred plants. The later are typical components of median deposits that are here found embedded in these dung laminations, possibly as a result of redeposition and trampling, or more likely to the fact that discard activities were not discontinued in this area during its use as a pen. Hence, in this particular case, the microcontextual evidence points towards the dual nature of this open space, and it's used both as a discard location and as an area of activity. It is, of course, possible that the alternation that we see between pen and median deposits is related to seasonal changes or to life rhythms within the site. So I'm going to leave it here. I'm going to thank all of these people that have been involved in my research, and all the organizations that have been funding my work, particularly in Central Anatolia, over the years. Thank you very much for listening. Alessandra, sì?
Thank you very much to all the presenters for the effort for keeping going on time, more or less. The last talk today is by Alessandra Pecci from the University of Barcelona. Okay. Hi, Alessandra.
Could you please share your screen? Yeah, you're mute. But no worries, take your time. Great, I can see. Let's try the presentation mode.
Can you see it?
Yes.
Oh, wait.
It's a slide presentation.
Yeah, it doesn't work. Okay, can you?
Yes. Yeah, I think the YouTube takes a little longer, but it's getting in progress. No, thank you very much for all your efforts to make it, because I know it's really tricky for many of us. Well, now let's move to the Mediterranean pack. And well, this talk is actually, it's a bridge between this late prehistory, protohistory, and the classic period. So it's much a bridge between these both sections. Classical archaeology session tomorrow. Well, that's it. Thank you very much, Alessandra.
Thank you very much. And thank you for the organization of this workshop that is really being interesting and very varied. So thank you for the possibility of being here at this workshop at the CSIC in Rome. And what I will present is something related with combining chemical residue analysis as activity biomarkers, archaeology of food production and consumption. Among the approaches that can make visible the invisible archaeological record, there is analysis of chemical residues trapped in archaeological materials. Residue analysis have been applied for over 40 years to the study of pottery and floors. And when I say about 40 years, I think of the works of the 70s that Luis Barba was carrying out in Mexico in order to study archaeological floors, and Condamine and Formenti were starting to develop the organic residue analysis of amphora in the Mediterranean area. But of course, since then, these approaches have improved, and many different laboratories are working with different techniques on the subject. So I will show some examples of these kind of studies, starting from the point that we can do them because liquid and semi-liquid substances produced during the performance of activities are trapped into the pores of the materials that come in contact with them and are preserved. Specific analysis must be performed, therefore, to identify the chemical residues absorbed by the porous material and interpret the activities that were carried out. As chemical residues absorbed by pottery and floors and their spatial distribution are strictly related to the activities carried out, they can be considered anthropic activity markers. As I said in at the beginning, I'm going to present data related to the study of the traces of food. So in order to study traces of food, we can analyze different types of archaeological materials. And in particular, we can work both on earthen and lime-crusted floors of buildings, and in particular of rooms such as kitchens, food production and consumption areas, etc. And then we can work on vats, floors, pottery of production installations, and on pottery used for food production, transport, cooking, preparation, and consumption. So different types of pottery that can be related with food processing. So the methodology we have to carry out every time we do analysis, scientific analysis applied to archaeological materials are, as we have seen the whole day, a first phase of sampling, then we do the analysis, and we have to choose different techniques in order to answer our questions, then depending on the samples we have, the money we have, the time we have, etc. And then we have a phase of interpretation that, as Manoni Giannichedda said many years ago, has to be done of two parts. One is understanding the chemical data in our cases, And the other one is understanding the meanings, the archaeological and historical meaning of these data, the scientific data. All these can be achieved only through an integrated approach. So we can study both floors and pottery. Depending on what we are studying, we can apply different methodologies so that when we work on floors, we usually take more samples in a grid, and maybe we carry out more simple analysis in order to be able to process a lot of samples. While when we study pottery, we usually go towards a more specific approach and techniques such as gas chromatography coupled to mass spectrometry in order to better identify the biomarkers of ancient substances that came in contact with pottery. So through the analysis, we can try to study something about the different phases of the life cycle of food, from its production to its storage, trade, food preparation, and food consumption. So I will show some examples deriving from different contexts that I've been working. So for food production, one of the themes that I've been working a lot has been the study of production installations for oil or wine. Why? Because although we might think that it's easy to recognize production installation for oil or wine because of their archaeological characteristics, is not always the case, and more even people that work in production installations say that it's more difficult than they thought to recognize them. But this is especially difficult when we have only the bottom of the vats that were used to collect either the must for the production of wine or the oil. So in the cases as the ones that are present in the slide, we really don't know how to differentiate an installation for wine or oil. So in this case, organic residue analysis can really help. And before trying to study the archaeological data, we always have to carry out experiments and ethnoarchaeological work or ethnoarchaeometrical work. This means the study of modern materials that help us better interpret our archaeometrical data that we are obtaining from the archaeological context. So in this case, in order to study food production installations, and in particular wine and oil, we carried out analysis of modern production installations. And in particular, the one that I'm showing here is one related to oil production. The study of this oil mill, this is from Mallorca, but many other studies have been carried out, shows that in the flows of oil production installations, fatty acids, in this case carried out, analyzed with a method developed by Barba at the UNAM in Mexico, are present in all the floor, mainly around the mill and the presses, but really all the floor of oil production installations is full of fat. That the GCMS analysis confirms that is fat that really derives from oil and not from contamination. So on one side, we know from ethnoarchaeological works that we can identify oil production installation. And on the other side, we also carried out a lot of ethnoarchaeometrical work with wine production, and also a lot of experiments, because actually when we try to identify wine, spot tests are not reliable, so we have to go straight to more precise techniques. HPLC can be used, or GCMS analysis. Different techniques have been developed. We are using a method we developed some years ago at the University of Siena, and we have working with this for many years, and we have republished it recently in the Journal of Archaeological Science. So having all this data allows us to go to the investigation of archaeological cases. An archaeological case that I love, because was one of the first ones that I faced, was the study of the so-called Villa di Augusto in Somma Breviana that was covered by the Vesuvium by the eruption of 472. So it's not the Plinian eruption of the Vesuvio. And here at the beginning, only some vats that are seen in the right part of the slide were excavated. So we sampled them, and in some samples we could identify the presence of wine. So we suggested that possibly this installation was for wine. But then excavation went on, and a cella vinaria was exposed, that you can see on the left side of the slide, and the dolia of the cella vinaria were exposed. So we could also sample these dolia, and we could confirm that wine was actually produced in this installation. And another case in which the whole installation was never found, and only the bottom of the one vat of the production installation was identified, was this case in Samesquida in Mallorca, where we took samples from this vat that was hypothesized to be used for clay decantation, because it was found in a place where also an oven for pottery was found. But actually the organic residue analysis allowed to confirm that this was used for wine production, because white markers were found and no contamination was there. So we could actually say that wine was produced at the site. So these are just two examples to show production of substances. And then we can study also storage of substances. And this is a case, a more ancient case, and at the Tuscan site of La Marsigliana in Tuscany, where the University of Siena and the Soprintendenza Archeologica of Tuscany carried out a project that was directed by Andrea Zifferero, which gave to light all these houses in which many amphora were found. So it was called the House of Amphora, but also many dolia had been exposed during excavation. So we sampled them in situ in order to perform organic residue analysis. What we found is that all of them are coated with pinace resin or pitch. This is something that is very normal for the Roman period, but we didn't know for the Tuscan period. So it means that the coating practice already existed in the Tuscan period, the coating now of dolia for the storage of substances. And again, we performed analysis in order to look for wine, oil, and other substances, lipids in general. So in some of the dolia, the ones that you see with a red spot, we identified tartaric acid and other acids that are considered markers for wine. And we can say that they are there because of the use of the dolia, because we also took samples from the stratigraphic unit, and we could confirm that no tartaric acid was in the stratigraphic unit, so it's not contamination. So we can suggest that at least part of the dolia were used for wine storage, maybe production, but we cannot say. Then two dolia, the two that have the black spots, did not display any organic residue. At least for the one on the right, we can suggest that it was used to store water, because it's connected by a channel to the impluvium of the house. So probably it was used to conduct the water, the rainwater. But there is another one that also doesn't display any residue. So we don't know whether it was not used, it contained water, or maybe it contained something that doesn't leave any residue that we can identify with the analysis performed. Actually, what we have to know is that we are looking for some markers. So we look for lipids, we look for wine, but maybe we should also look for something else, or there are markers of substances that are still not recognized. So we are not seeing everything that was contained in ancient potteries. And finally, you can see the green spots. The rest of the dolia were probably used to contain a fatty substance that is compatible with olive oil. It's difficult chemically to assure that it was olive oil, but let's say that it's compatible. And to go further in the investigation, we also took samples from the floors. Unluckily, we were not able to take the samples of the whole house, and we concentrated in the part that you see in the map. So here we did phosphate analysis, again with spot tests developed in Mexico. And what you can observe, if you see, there are darker colors on the right and on the left. This is the open air part of the house, not the exterior part on the right and the courtyard on the left. This means that the external parts are more enriched in phosphates. So probably more dirty activities were carried out in the outside than in the inside of the house of storage, whatever it was, the house of the emperor. And we also did the analysis looking for fatty acids, again with spot test. And what we can see on the map is that there are fats that probably could be related with the storage of oil, because we can see them around the pottery that contained oil, that is the one that is marked in green, and that was found broken on the soil. So probably when it broke, it was full. So the fats that it contained also enriched the floors. So this is a way that it's possible to combining organic residue analysis of pottery and floors in order to better understand the use of space and the activity performed. Then I could not resist and also show the study carried out at the Garum workshop together with Dario Bernal Carrostola and Daniela Coptica in Pompeii, in which again we tried to combine the chemical study of the floors and pottery. So it was not possible to sample the whole area. This is a Garum workshop. And this is very well known because in the Dolia, fish bones were found. So it's very well known, and the University of Cadiz has performed wonderful study of the fish bones and the pollen in order to study all the preparation of the fish byproducts. But what we did was sampling the floor under and around the Dolia, and we found that, again, we can see a lot of phosphates, that it's enrichment in organic matter, and also fatty acids that in this case were probably related to the spilling of garum that is also rich in fat. And we also did the analysis of the Dolia, which had fish bones. So we tried to figure out if there was something else. And we identified all the residues of the coating. So peach again, they were all coated as the ones from the Marsilliana, but they also display wine residues. So here come the problems. Was this a reuse of Dolia that were originally used for the production of wine and then reused for fish, or wine was used for the preparation of some kind of fish byproducts? We cannot know because we don't see the stratigraphy of the absorption. And we also studied amphora that were covered in situ. These are two amphora that are supposed to carry oil. One is an African amphora, and the one on the right is a Dressel 20 amphora coming from south of Spain, from Bética. So both these amphora display the residues of the possible oil that they had originally contained, but also display a reuse. In fact, in the African amphora, lime was found, lime that could have been used to reduce the acidity of the fish byproduct production, as University of Cadiz suggests, or as a building material, as whole Pompeii is full of lime everywhere, because they were actually restoring the houses and buildings after the earthquake of 62 after Christ. But these data suggest us that amphora were also reused. They probably arrived at Pompeii carrying oil, but they were reused with something else. So the African amphora with lime and the Dressel-II amphora, either to put olives if they were not the original content of the amphora, but to put stones of fruit, well, but to put fruits because a stone of Prunus avium was also found. So this takes us to another subject that we're going to treat, that is amphora and all the problems of interpretation of content of amphora that can arise. So amphora have been studied during a long time in order to better understand ancient economy in the Mediterranean area mainly. And traditionally, specific typologies have been considered suitable for specific content because of their form, because of their provenance, because tituli picti that speak about the content have been found, or because solid contents or stamps have been found. So they all suggest the content. But unluckily, this is not always so straightforward and some problems can arise. So organic residue analysis have been used a lot and mainly during the last years in order to try to better understand the content of amphora. I will quickly show some examples. One that is quite useful for us because it really confirms the archaeological hypothesis is the study of Dressel-20 amphora from the Testaccio in Rome that actually confirmed the presence of oil coming from the Betica. But what we also identified was an organic coating, again, made of pinnacle products of these amphora. And it had traditionally been said that oil amphora were not coated. But during the last years, all the analyses that were carried out from different laboratories, Nicolas Garnier, Romanus, York University, etc., we have all shown that amphora were often, almost always coated, not depending on the content. And then, so this is another thing that organic residue analysis has discussed, not the presence of an organic coating or not. Another project that allowed the confirmation of ecological hypothesis is the project carried out at Oplontis in collaboration with the University of Texas, in which we could analyze a Dressel-II for amphora of Vesuvian production that were supposed to be wine amphora and actually carried wine. Then it's more complicated than this, but let's say that at least wine was present in the amphora. But then during the last years, we had also problems. So in some occasions, we have found that contents different than those expected were identified. The first example I have in mind is the castor oil present in some amphora recovered at Classe that was actually told us that amphora could be reused or in some occasion could have a content different from the one that we expected. And then other problems are emerging now that we are carrying out a project financed by the Spanish Ministry of Science and Innovation, studying Roman amphora in order to identify the content. And we are doing a work in Pompeii together with Alessia Contino, studying the organic residues of amphora. She had already studied for her PhD of Northern African amphora that are supposed to carry oil or olives. And actually the results that we have until now is a mixture of substances. We have sometimes oil, but we have mostly wine. And most of the time we have both. So this is a problem, and we are trying to figure out if the amphora arrived with oil or olives to Pompeii and were then reused, or if they arrived with wine and they were reused, or if they, or it's possible that they were containing a mixture of substances such as olive in vinegar or wine. So we're doing experiments. So this is an example of the problems that we are facing. So quickly through food preparation, in order to study food preparation, we can study kitchen. So again, at the beginning, we studied known cases as the one of San Vincenzo Volturno, in which we had wonderful kitchens from the monastery, very understandable. So we took samples, we did the analysis again here, the spot test, and we can see to the left phosphates unenriched around the fireplace and the ovens. We have also compounds related to protein residues around the fireplace and fatty acid in the kitchen. So with this pattern in mind, we went to study other case studies. One was San Genesium, a building made of 2 rooms. It's a medieval building that was destroyed in 1248 by a fire. And what we have here is, again, we did spot test. If you see, the whole floor is dirty, is full of phosphates and fatty acids. This is not compatible with the possibility of having one room for food preparation and another for sleeping that doesn't leave any trace. So what happened here is that they were cooking and cooking and cooking and making the floor dirty, you know, with all these residues of fatty acids and phosphates. And this is because we afterwards understood that this was actually a tavern that was located along the Via Francigena, where pilgrims probably stopped. So what we are seeing is kitchens preparing a lot of food, and in which the other archaeological indicators helped and gave us the answer to our question of why we had so many residues, because jugs with wine were found, and money and dyes. So all this together allowed us to have this interpretation of a possible medieval tavern. And in this case, we also took samples from the area around the fireplace. We analyzed them with spot tests, so just the first step of analysis. But what we could see is that not only the floors, but also the pottery, of course, was full of phosphates and fatty acids. And also the testi, that are ceramic form used apparently to cook bread, in some occasions also displayed fatty acids. In these cases, probably they were either used to cook bread with some fats, or possibly they were used to serve food or something else. So a difference in the use of the testi was suggested. So in this case, we could not go further with GCMS, but we could do this at another site that is Donoratico, again a medieval site excavated by the University of Siena, in which there was a hut with an earthen floor and post holes with a fireplace. So we took samples around it, and we can see that around the fireplaces there are residues that indicate the preparation of food. We don't have residues inside the fireplace because probably the heat of the fireplace destroys them, degrades them, but we have all around. So there was food consumption and preparation area, and we also did the analysis of pots and testi found in the same context, and both with spot tests that indicate the presence of phosphates, fatty acids, and these compounds related to protein residues, both in the pots and the testi. And we could confirm this with GCMS that confirmed the presence of animal origin and plant origin fats in the pots of Brassica oleracea, that's cabbage, that it was very common in Tuscany during the Middle Ages. And in the testi, again, we could identify fats that come from animals. That also suggests, as in the case of that we have just seen of the medieval taverna, suggests that testi were not only used to cook plain bread, but probably either fat was mixed to the bread or they were used as serving plates or something else. So again, it's nice to see how the combination of pottery and floor study can help us better seeing the traces of kitchens. Then quickly through consumption, it's also possible to study pottery used for consumption. Very quickly, I'm showing here a jug, medieval jug used to serve wine, and a catino that was used to serve the same food that we actually usually find in the pots used to cook the food. And then we jump to a much more ancient case study to see consumption of wine that is much older, that was recovered in Pilastri di Bondeno e Terramare site, investigated by the Soprintendenza Archeologica and the University of Padova. That allowed us, through a very serious and delicate integrated approach together with pollen study, botanical study, etc., to suggest that already in the Bronze Age, in these Terramare sites, they were starting to consume wine, and we don't know whether even producing them. So with this just last example, I leave you, but just telling you the last thing that I think is very important to remember when we do organic residue analysis, that is that porous materials absorb all the substances that they come in contact with. So what we find is probably a sum of substances, and the problem is succeeding in identifying them, separating them, and trying to understand what comes first, what comes after, and if it's a mixture, which can be done only studying the archaeological context. So thank you all to have let me given you some hints of the possibilities of the analysis of organic residue analysis applied to archaeological materials. And thank you very much again. Okay.
Thank you very much, Alessandra, to make it on time. Maybe we have very few minutes.
I'm trying to see if
you can stay here a little more for the discussion right now.
Yes.
Yes. Any questions? Well, it was really amazing, all the examples.
Yeah.
Case studies is really well. Thanks a lot. I don't know if there is something, there is a comment in the chat, but there's not a question, it's just a general comment that you have now something related to Alessandro, other speakers. I'm sorry, I think I missed my microphone. Okay, so here again. Well, now we have little time for some discussions, questions, or whatever. No, just very long session.
Yeah.
We have
very
things that were common with some of the talks, especially you mentioned lots of challenges and issues that we are facing when creating different methods. That don't know if something.
Sorry, I cannot hear very well. What did you say?
Yeah, no, regarding the challenges that you mentioned, some of the issues that you're facing when especially with combining these different perspectives. And I mean, well, you mentioned some specific issues, but just I have my notes in the other side of the table now.
Oh, yeah. Well, what I haven't mentioned but should have said is that actually integration should have carried out not only with between different organic residue analysis, but all the studies that you have shown during the whole day are absolutely super important, and luckily more and more they are combined, no? Micromorphology, starch, phytoliths, and everything is really important. And for the study of pottery, I also think is very important, because although in some cases cereals can be identified, is not reliable. Organic residue analysis, I think, is much better to carry out microbotanical studies. And also for the study, the use of flows, etc., we have been doing with Marco Madella, Carla Lancellotti, some experiments of combining data that show the importance of doing this with Marco as well. And I think that also the new studies that have been carrying out with XRF analysis on flows, that now is like a boom, should also be integrated in order to really see how they are complementary or not. So I really think there is much more to do.
And also, from the experimental side of view, because we have many examples this afternoon on combining this experimental data, which is really critical for us. You mentioned your ethnoarchaeometrical work, or how you call it? Ethno-experimental, or no?
Ethnoarchaeometrical.
Ethnoarchaeometrical.
It's a definition that was used at the beginning for the production of pottery. Busheda cow started to use this for pottery production, but as I come from Italy, where organic residue analysis is archaeometry, not necessarily bioarchaeology like in Spain, or at least in Catalonia, I'm used to think about these studies as archaeometry. And I think that actually it is an ethnoarchaeological work, but it's really aimed at better understanding the archaeometrical or scientific data. So I like the definition of ethnoarchaeometry.
So we use the definition of geo-ethnoarchaeological work too. So this combination is very funny. Yeah, we have something if you want to comment. Yeah, because there is an issue in the chat that was related to your talk. So maybe.
Hi, Alessandra. I would like to ask a question, if that's okay. I'm really interested in GCMS as well, and I was wondering whether you have ever experienced any problems with contamination of samples, because, for example, I'm aware that plastic can really distort results, if you touch the sample with your hands, we have cholesterol in our fingers, and that can also cause a peak in cholesterol. So what are your experiences with this, and what advice can you give us?
As you said, the best is not touching and not doing anything. Plastic really can affect, and it's really visible. The problem is that sometimes, as you have just said, the peaks of plastics, of phthalates, are so high and so abundant that it can cover the archaeological substances, no? Because the traces are much smaller, and depending, some of them can be covered. So plastic can be a problem, it should be avoided. I always use aluminum foil, and wherever I can, I use aluminum foil for anything, even in the sampling. Cleaning the surface of the ceramic in order to take away the possibility of the skin, no, to have left some residues. And what we also always do nowadays, at the beginning we didn't, because we didn't think about this, but during the last many years we have been doing this, is taking control sample from the stratigraphic unit. That I think is very important. Now, mainly when there is a discussion is what you have found really there, or does it come from post-oppositional contamination? It's important to be able to show that it's not in this stratigraphic unit, but in the sample itself. But yes, contamination is a huge issue. Even in the lab, you can have contamination. So you have to check it. Every time you pass a sample, you pass controlled samples to verify contamination. It's a huge work. We never leave our material in the lab. We always take it home in order to be sure that nobody uses them, so that they're not contaminated. But yeah.
Thank you. So I don't know if there's any questions for Julia, Bailey, well.
Yeah, I have a question for Julia. Thanks for the interesting presentation. About linking the different things. So if I remember, there was a lot of information today, brownstone tools, yeah, and starfish, and identifying new structure, like micro remains. And so, is there also pottery assemblage, maybe food first, so that the food make the next step and see what kind of food products.
Sorry, Julia, could you come here and repeat the question and so on?
I can repeat the question.
Thank you so much.
She was saying that I presented it was quite informative in terms of starch from ground tools. So possibly if we can have the same or different kind of data coming from pottery as residues. So this is a big thing. I mean, the group of Bristol, University of Bristol, led by Richard Evershed and especially by my colleague and friend Julie Dunn, worked extensively on these in the framework of the Green Sahara project. And basically, they collected all the possible pottery assemblage. So they have, at the moment, even if they is still under study, the assemblage from Awaftea. So from the site that I presented, we gave them more than 100 pottery sherds that were kept at the Museum of Archaeology and Anthropology at the University of Cambridge. And so we are waiting for the result. For Egypt, unfortunately, we don't have anything for two reasons. First, because the site that I presented, that there is almost no pottery. Possibly we found 5 sherds in more than 10 years of research. And this was another point I didn't touch, but it's very interesting. A lot of ostrich eggshell used also for cooking, because we found evidence of burnt sherds. So possibly some of them were definitely used as bottles, but some others also as a sort of a bowls for cooking. So this is the first point. And the second point I mentioned during my talk, that there is no possibility to do this kind of analysis in Egypt. So you cannot export materials. So actually, whatever you find in terms of archaeological science related to Egypt is always done on museum collection outside Egypt. So this is really, there is something that has been done, for example, on the Neolithic site of Merinde, but again, pottery sherds kept in Sweden. So it's really a pity, but I mean, doing especially prehistoric archaeology in Egypt is sometimes it's frustrating, because it's doing like Flinders Petrie used to do it 100 years ago. So, yeah.
Thank you.
Thank you very much. I think I don't have any questions in the chat right now, and I think it's 7. let's continue tomorrow. Tomorrow, the session on classic archaeology. Well, we had a really nice introduction by Alessandra Pezzi today, and medieval archaeology also. And that's 9:30 tomorrow, so same place. Thank you very much to all attendants and for your patience to be here, and hope to see you all tomorrow again. Thank you. Mm-hmm.